So what supports this particular thing? What supports that evolution happens? See, cytoplasm, when we talk about cytoplasm, inside the cell, okay, let's say this is the cell. Inside the cell, there is a jelly-like fluid, okay, jelly-like fluid. Diagnostics, development of PCR-based test for detecting diseases like COVID-19. So it's diagnostic. Diagnostic means identifying whether a person has disease or not. All right. So basically, like yesterday, we have discussed about space science and technology and physics. So we have discussed many topics yesterday. I mean, like one topic was remaining, which is printed in the book. Okay. So I would just suggest you to go through that topic. You will be able to understand that topic based on the discussion that we had yesterday. All right. And so maybe in the last day, I may take a combined class of many topics from multiple areas. Okay. So as of now, we are going as per the plan. Whichever day, whatever is planned, we are going to take the discussion over there. Whatever additional topics will be there, there can be an additional class, or let's say I'm going to give you the notes in printed format. Okay, so that way we are going to cover the things. So basically today we want to discuss about biology and biotechnology. All right. And so we are going to discuss those topics that are important for the examination. Okay, in such a manner so that your conceptual clarity about these topics are clear. Okay, and whenever let's say you feel that like you need some additional clarification on certain topics, we may definitely discuss about that. Okay, so the thing is like the first topic that we are going to discuss today is, let's say, the topic number 1 is theories of evolution. Okay, theories of evolution. Now the question arises, what do we mean by evolution? Okay, what do we mean by evolution? So when we talk about evolution, it is like how the species evolve over a period of time, how they change, right? So we know heredity and evolution. Heredity means when genetic information is passed from one generation to the next generation. So when genetic information is passed, I mean like, so there are some genetic mutations also. I mean like the daughter cells may not be the exact replica of the parent cells. I mean like, there may be some changes over there, right? And so the thing is, when we talk about heredity, means inheritance, or let's say carrying forward the genetic information from the previous generation to the next generation, that is inheritance. Then evolution is like during the lifetime of an individual, right? So what changes are happening in his or her bodily structure and all, it depends on certain environmental let's say factors. I mean like some species are able to, let's say, adapt themselves to certain environmental conditions, and if these adaptations are beneficial for them, so it is going to help them to reproduce over a period of time, and then they are the ones who are going to be the fit for the, let's say, like environment, and they are going to survive and they are going to reproduce again. Okay, so today basically like we will discuss about theories of evolution. So related to theories of evolution, there have been many people, right, in the past who have given let's say a lot of ideas related to evolution. All right, so basically like when it comes to theories of evolution, right, some prominent names are like you might have heard about Charles Darwin. Okay, Charles Darwin had talked about evolution also. So he talked about principle of natural selection. Okay, principle of natural selection also. So we are going to discuss about many things today. So first topic, quickly write down the name of the topic, which is theories of evolution. Write down theories of evolution. Or you can write down definition of, like this is the topic, now write down definition of evolution, so that like the concept is clear to all of you. Definition of evolution. So write down, evolution is defined as the change. Evolution is defined as the change in heritable characteristics of biological population. In heritable characteristics of biological populations over successive generations. Over successive generations, right? Now write down, it occurs it occurs when evolutionary processes such as natural selection and genetic drift act on genetic variation within a population, leading to changes in traits that become more or less common over time. Okay. Now we are going to discuss about a prominent personality. His name is Charles Darwin. So Charles Darwin talked about principles of natural selection. Basically, he had written a book. Its name is On the Origin of Species. Okay, what is the name of the book? On the Origin of Species. Okay, on the Origin of Species. So basically, when we talk about this particular book, here he discussed about principle of natural selection. What he discussed? He discussed about principles of natural selection. So we will discuss what do we mean by principles of natural selection. Principles of natural selection. So when we talk about principles of natural selection, so it is a three-stage process according to Charles Darwin. Okay, it is a three-stage process. So what are those three stages? That, you know, so individuals within a species exhibit variations in traits. What do we mean by traits? Yes, individuals within a species, right? Individuals within a species demonstrate, right, variation in traits. They demonstrate variations in traits. Now the question arises that what do we mean by species? Okay, what do we mean by species? So basically, like, you know, today itself we are going to discuss about classification. I mean, like taxonomy, we are going to discuss about it. So we are going to discuss like how organisms are classified, okay, how a classification of organism happens. Okay, so we will discuss about, let's say, like, you know, like three classification system, and then we also have, let's say, like, you know, 5 model classification and 6 model classification. So what do we mean by species? Species, the lowest means, like, you know, a species is a similar type of, let's say, individuals. Okay, so one particular snake may belong to a particular species. Okay, bird is a type of a species. So, like, you know, they are the lowest level. Lowest means, like, you know, most distinguishable. I mean, like, you will be able to categorize one species from the another one. So basically, like, in a particular species, let's say, like, you know, there is a species of a snake. So all of them are snakes, but, like, you know, there can be variation within those snakes. Okay, there can be bird species, I mean, like, similar bird species, there can be variation in them. So basically, Charles Darwin, what he is saying, that individuals within a species, okay, inside a species, individuals may show some variation. I mean, like, some may have longer nose, some may have shorter wings, and also this kind of variation will be there. Okay, so individuals within a species demonstrate variations in traits. Okay, traits means visible characteristics, anything that is visible to us. Okay, visible characteristics in let's say individuals. So this is one important thing, that, like, you know, individual within a species demonstrate variation in traits. Then second observation that Charles Darwin made, he said some traits confer advantages in survival and reproduction. Okay, some traits confer some traits confer advantages. Some trait confer advantages in survival and reproduction. Okay, basically like survival and reproduction. Now what do we mean by this? Basically, I mean, like within a species also there can be some trait in individuals. I mean, like if you read the NCERT books, so in NCERT books they have given a lot of examples. For example, let's say like there is a species of beetle. Okay, beetle. Many beetle are let's say like green in color. Some are red in color. The ones that are red in color, so they are visible in forest. Okay, so those species that eat upon those beetles, they'll be able to identify or spot those individuals, so they'll eat them away. Then the green ones, they're naturally fit for the environment because the environment is already green. I mean, like all the forest, their color is also green, so like they are not visible. Okay, so the thing is, the birds or who eat upon them, so they will not be able to identify them clearly. So this particular trait, which is trait means characteristic, like change in color, I mean, like that actually helps, I mean, like the green ones to survive more. So this particular change in color, I mean, like they are similar with one another. One is green color, other is red color, because that is red color in green forested area. So red is spotted, can be identified easily, so that gets eliminated easily. The green ones, I mean, like since they are not visible, I mean, like to the preys, okay, so like they, I mean, most of them will be able to survive. So this helps them to survive and this helps them to reproduce also. So he is saying that some traits confer advantages in survival and reproduction. Okay, so these are natural traits in individuals. Okay, and those species that that have advantageous traits, they are the ones who are going to reproduce and who are going to, I mean, like who are going to survive. Their generations are also going to survive. So this is, I mean, like principle of natural selection. I mean, like naturally some species, I mean, like some individuals of a species may survive, some may not survive because of visible traits, because of, let's say, natural trait. Okay, so here I'll write it clearly: principles of natural selection. Principles of natural selection. So what is the third point that he says? What is the third point? He says that individuals with advantageous traits are more likely to survive and reproduce, passing those traits to their offsprings. Offspring means their children, basically, right? Their younger ones. So like these individuals, I mean, like these traits are basically what these traits happen. These traits are passed on. Okay, these traits are passed on. Write down this line, third line, quickly. Write down: individuals with advantageous traits. Individuals with advantageous traits. Individuals with advantageous traits, right? Write down: are more likely to survive and reproduce. Are more likely to survive and reproduce. More likely to survive and reproduce, passing those traits to their offsprings. Okay, passing those traits to their offsprings. Offsprings means, all right. So what we have learned now, we have understood about Charles Darwin. He had talked about principle of natural selection. Natural selection means like nature itself, I mean, like helps certain species to survive, and nature itself, let's say, like decides that which species is not going to survive. Okay, this is the thing. I mean, like we have talked about beetle. There can be many similar, I mean, like, beetle green color and few of them may be red color. So red colors will be eliminated because like birds will be able to spot them, will be able to see and will actually eat them away. But the thing is like the green ones, they have more chance to survive because naturally some traits are beneficial for species. So the traits that are beneficial, these traits are going to help those species to survive further, okay? And they will be reproducing and these traits will be passed on to the next generation, all right? So is it understood?
Yes.
Now, here tell me one thing, okay? So I'm just going to ask a question. So what do we mean by evolution? One person will raise hand and then he will answer. Yes.
According to changes in environment, species adapt certain traits and accordingly their evolution happens.
Mm-hmm.
Their adaptation is matched with the existing environment that helps their survival.
Mm-hmm. Mixed up facts little bit. Yes. Anyone else?
So,
Yes.
so adoption or modification with regarding to challenges or difficulties in their survival.
Adaptation or modification with regard to
challenges. Survival.
its survival.
Yes.
Okay. So these adaptations and modifications will help them to evolve. Okay. Over a period of time. Okay. So that is evolution. Yes. Sri Naiki.
Change in characters over time, sir.
Change in characteristics. Okay,
Over time.
generally like we know that like over let's say like generations to generation, I mean like inheritance happens, heredity, I mean like the newer generation, they inherit the characteristics of their parents and then they will pass on their own characteristics to their offsprings, right? It keeps on happening. But the thing is like some species, I mean like some species basically adapt to the changing environment and all. So that's why he also talked about, let's say, fittest, like survival of the fittest. I mean like the one which is which adapts to the changing scenario will be able to survive. Others will be wiped out, and naturally it happens. It happens, let's say, like in the let's say it happens in nature also. It happens in other parts of life as well. If you talk about businesses, right? People who start businesses, you might have heard about the startups and all. So a lot of startups are started by people, but some are able to adapt to the changing requirement, right, from time to time. And the ones who are going to adapt to it, they are going to survive their business. Okay, they'll be able to survive in the long run. The ones that are not able to adapt, the ones that are not able to understand what is needed, what is not needed, they will not be able to survive. That's the thing. So I mean like it is applicable everywhere, but here we are talking about biology. Okay, so this is, I mean like we have discussed about evolution. Now what do we mean by let's say like principle of natural selection?
Species adapt various kind of traits and whichever is more suitable to the environment, that character leads to their survival in the
Okay, one person will tell me one point, the next point will be told by the other person. Okay, so principle of nature. So the question is, what is the name of the book written by Charles Darwin? On the origin of species, right? On the origin of species. Now what was the first point he said on principle of natural selection?
Variety in same species. Varieties in same species.
individual trait.
There can be variation in traits in a species, individuals of a species.
One species.
Yes, yes. There can be variation in trait. For example, I talked about color, changes in color. One can be green, other can be red. Any other example that you can think of?
So, tigers can walk on go on trees and lion cannot. So means
Like birds, bees.
cat family से दोनों हैं. लेकिन
So
पेड़ पे चढ़ पा रहा है एक नहीं तो
एक नहीं चढ़ पा रहा.
तो सर्वाइवल हो पा रहा अगर टाइगर कमजोर है.
हाँ. That's why you mean to say कि हाँ जो lion है, ठीक है, मतलब like they are,
एक एडवांटेज हो जाता टाइगर के पास.
हाँ हाँ हाँ,
Like
ठीक है.
breed of birds, बड़ा बड़े बड़े birds जैसे चील उसका बड़ा भी होता है वो एक करता है उसको.
All right. What is the second point? Quickly.
Some traits are advantageous.
Yes, some traits are advantageous to individuals, right? Third point is these advantageous traits, I mean like individuals with advantageous traits will be able to pass on, I mean like will be able to survive and reproduce and pass on to the next generation. Okay, so we have learned the first point. Now let us discuss, so we have already discussed about natural selection. This is known as principle of natural selection. Now we will discuss about another thing which is known as genetic drift. Okay, we are going to discuss about another thing which is genetic drift. Okay, quickly. Genetic drift. Okay, I mean like very less number of these things are there, so you will be able to understand them clearly. Quickly write down about genetic drift. Write down so that you can remember these points. Okay, so write down genetic drift refers to random changes in allele frequencies. Genetic drift refers to random changes in allele frequencies. Genetic drift refers to random changes in allele frequencies within a population due to chance events. Due to chance events. Full stop. This effect is more pronounced.
So please repeat random shift in.
Okay, I am repeating this genetic drift. Genetic drift refers to random changes in allele frequencies. Random changes in allele frequencies. Quickly write down. Within a population due to chance events. Within a population due to chance events. This effect is more pronounced in small populations. This effect is more pronounced in small populations and can lead to significant evolutionary changes over time. All right, let us understand about this. Okay, what do we mean by genetic drift? Now here, if I talk about genetic drift, what is happening here? I mean like drift, we understand. What do we mean by drift? The keyword drift. Huh? हाँ हाँ.
So drift refers to carrying forward something, carrying forward something.
Okay.
Sudden change.
Sudden change. Continental drift theory. अच्छा, हाँ. Yes. हाँ हाँ. Tell me.
Actually, the change in path direction जो आप जा रहे थे.
हाँ हाँ हाँ. Change हो गया. Drift. All of a sudden. ठीक है. So let us understand about this particular thing. So what is happening? We are talking about genetic drift. Genetic means like genetic means it is associated with genes. Genes means these are those things that carry the information how the individual is going to be. I mean like when reproduction happens from one generation to other generation, this genetic information is passed on. This genetic information helps, I mean like the other individual to, I mean like carry forward all the information that their parents had. I mean like if their parents are human beings, so the child will be human. I mean like he will be having 2 hand, 2 leg, and all these things will be there, and they will be identical also. Now genetic drift, I mean like changes in the genes. Okay, here what is happening? Changes in the genes. I'll give you an example. If I talk about eye color, I mean like the black ones that we have, right? Some people have it black, some people have it brown, some people have it blue also. I mean like eye color, some people have black, brown, blue. I mean like some people may be having green as well. Okay, so why it happens? Because the thing is like the genes that make up the eye, okay, They are multiple genes. It depends which ones are combining together and which one is the dominant one. I mean, like for example, you heard about blood group also. I mean, like blood group, all of us have different blood group. I have O positive, some of you may be having AB positive, A positive, B positive, like that. Okay. It depends like which particular gene, I mean, like, when we talk about genes, which particular gene is dominant, which one is recessive. Okay. I mean, like when dominants come, one dominant, one recessive is there. So these are basically known as alleles. Alleles means like different form of the same gene. Okay. The dominant one will get expressed. The recessive one will not get expressed, but it will be available there. Okay. So what happens here when we are talking about, I mean, like, we know that, like, mutations happen. Mutations means like changes in genes happens, right? So over here what happens, the allele frequency, for example, let's say, like, an individual has multiple types of genes, I mean, like same gene of multiple variants. And then those variants, I mean, like if there is a small population means, I mean, let's say, like there can be a particular, let's say, species for which the population is large. There can be species for which a population is small. Now, if let's say, like, some allele frequencies are changing, okay, means like, some allele frequency are changing, it is more likely that this particular population, I mean, like, that had less number of individuals. So they will have a genetic drift, means like all of a sudden after some time, I mean, like the entire next species, I mean, are going to come. So they may be entirely different. Entirely different means like many of their characteristics will be totally different as compared to their previous generation. So that is known as genetic drift. Okay. For example, I'll give you example. For example, if I talk about birds, if I talk about reptiles, I mean, like many of these things have like feathers that have similar bone kind of thing, similar bone skeleton. Okay, I mean, like bone structure is same, but the thing is their function has changed over a period of time. Okay, their function has changed. So it happens because of changes in the allele frequency. Allele means like, different forms or of the same gene. I mean, like I talked about the eye color. Okay, some of them will be recessive, some of them will be dominant. If there is a dominant, I mean, like one recessive, one dominant will get expressed, and both are recessive, then recessive one will get expressed. For example, in India, we have less number of people who have blue colored eye or green colored eye, maybe. Generally, like we have either black or brown, maybe. I mean, like, black or brown. Generally, black is more, brown is little less, blue is once, I mean, like, very less. Ashwarya Rai. Okay, I mean, like entirely different. So that's the thing. But the thing is, like, this feature is more common if we go to Russia. I mean, like there people, I mean, like, it is more common. You will find more number of people with blue colored eyes like that. Okay. So it is, I mean, like, within population, how those changes happen. Okay. So this is known as genetic drift. Okay. Concept of genetic drift is associated with mutations or change in how the allele frequencies are expressed over a period of time. Are we able to connect with this? देखो एलएलई फ्रीक्वेंसी मींस जैसे मैंने बताया कि मतलब आई का कलर है, मतलब रॉड शेप्ड सेल्स होते हैं, ठीक है, सम सेल्स आर रिसेसिव, सम सेल्स मतलब मान लेते 4 है, इनमें से कौन कौन से कॉम्बिनेशन आई मीन लाइक फॉर्म करेंगे, दैट इज फ्रीक्वेंसी, लाइक कि कौन कौन से आएंगे, कौन कौन से वो करेंगे. सो दैट इज एलएलई फ्रीक्वेंसी में जैसे जैसे चेंज होगा, छोटा पॉपुलेशन इट इज गोइंग टू सी जेनेटिक ड्रिफ्ट मोर फ्रीक्वेंटली, मतलब बहुत जल्दी में होगा. बड़े पॉपुलेशन में इट इज नॉट गोइंग टू हैपन एज सून, बिकॉज सम इंडिविजुअल्स विल सी सम चेंजेस, बट लाइक more number of individuals will be similar as the previous generation. That's the thing. Okay, so this is known as genetic drift. Write down next point, mutation. Mutation is a very important concept here. Okay, fourth point, sorry, mutation. Write down about mutation. Mutations are random changes in DNA sequences. Mutations are random changes in DNA sequences that can introduce new genetic variation. That can introduce new genetic variation into a population, while, full stop, write it down so that you remember, okay? Mutation, I am repeating, mutations are random changes in DNA sequences. Mutations are random changes in DNA sequences DNA sequences within a DNA sequences that can introduce new genetic variation, into a population. While many mutations are neutral or harmful, some can confer advantages that can enhance survival and reproduction. Okay, I mean, like, mutation means जीन में मतलब लाइक DNA sequence में changes हो सकते हैं. I mean, like, maybe base pairs are connecting differently. I mean, like, in human beings, like this year, if you have read about the Nobel Prize, okay, Nobel Prize. So basically, Nobel Prize has been awarded. I'm talking about 2024. For 25, I mean, like, maybe in the month of December or October, I mean, like, it is announced. Okay, October or December it is announced. Just a minute. So we were talking about mutations. So generally, like, I was talking about Nobel Prize, right? So what has happened? I mean, like, Google's DeepMind, they have created a system, right? Using which what we can do, we can identify structure, protein structure. I mean, like, they have created a model, artificial intelligence model. If you have DNA sequence, okay, DNA sequence of anything, right? You just put it there, and they are going to tell you what is the protein structure. If you give the sequence, it is going to give you the protein structure. Earlier, this particular thing was one of the let's say, like, one of the most difficult tasks to identify what is going to be the structure for a protein if we have the sequence of it. Okay, so, like, what is going to be the protein structure there? I mean, like, it used to take decades to identify protein structure of a particular protein sample. So the medical fraternity was facing a lot of difficulty related to this, but they have developed a system, okay, artificial intelligence system, and where, I mean, like, all the known proteins of humanity, I mean, like, whatever known proteins are there, using that AI system, they have been able to identify their protein structure. Okay, and if you have a protein structure, like, if you put it there, so it is going to tell you the DNA sequence. Okay, so it has revolutionized the field, and that's why these people have been awarded with Nobel Prize. Okay, they have been awarded with Nobel Prize in 2024. In fact, Nobel Prize in 2024 in multiple fields, whether it is biology, whether it is chemistry or physics, all of these things went into artificial intelligence related activities. Okay, so if you read it, so you will find ki artificial intelligence. Why? Because these people have created those systems. Okay, earlier it was very difficult. I mean, like, if you have the protein sequence, it is not necessary that, like, they are going to form into this kind of a structure. Okay, so there are many computational things also there. So it is very important for us to understand that DNA sequence, I mean, like, you may be having the same base pairs, but it depends how they are sequenced together, how they are connected. Okay, how they are connected, it will determine what will be the structure of it. Okay, it will determine. So that's the thing. Now, when we talk about DNA, so DNA is something that has double helical structure. I mean, like, it has a structure like this. Generally what happens, and like on one side we have some other base pairs, on the other side we have other base pairs. So when we talk about DNA has three important things. One is base pairs, second is the sugar, third is the phosphate group. Okay, these are the three important things that basically determine DNA. So it depends how they are connected with one another. Okay, how they are connected, it determines their structure and functionality also. Functionality also changes how they are connected. So basically in DNA we have the sequence which is like ATGC, okay, ATGC. Adenosine, thiamine, guanine and cytosine. Okay, in RNA we have AUGC, which generally has single helical structure. Okay, it has double structure and they have single helical structure in RNA. So like DNA sequences, we are going to discuss about the DNA sequencing also today in detail. We are going to understand about difference between DNA and RNA. Okay, so we do have a proper article based on that. Okay, so we'll discuss. So here when we talk about mutations, mutation means random changes in the DNA sequence that happens in individuals within a population. Then it is going to, let's say, create changes in those species. So this also leads to evolution over a period of time. So we are discussing what leads to evolution. Evolution means changes in the traits over a period of time. It may happen because of, let's say, mutation also. Mutation means random changes in the DNA sequences. That's the thing. Okay, so what supports this particular thing? What supports that evolution happens, right? If you read the 11th standard book, I think geography book, they have discussed about this thing in detail. Huh? Ultraviolet rays
cause mutation, genetic mutation.
It may cause, maybe in some species, it is generally harmful for, for human beings. It
Yes.
causes a skin cancer to us. Okay. Basically, UV rays are used, right, for medical devices, for cleaning or disinfecting the medical devices. Okay, so doctors who are doing surgeries, operations and all. So after doing a surgery, those equipments are very expensive. So they clean up those equipments. After cleaning, they put it through the UV rays so that it is completely disinfected, so that the same instrument can be used once again in the next surgery. Okay, in big hospitals, they have the facilities and all. So UV rays are different, so we'll discuss about this. So now fossil record, okay, what happens? So if you have studied about this, what we say, the 11th standard book, so it says when we talk about continental drift theory was there. So one big continent was there, Pangaea, I think, right? So, okay, Panthalassa, Pangaea and all. So like in the middle of it somewhere rupture has happened and then they drifted. Drifted means they parted ways and went somewhere else. But the thing is if I talk about in one particular continent, okay, maybe over here if you find some species, okay, and over here if you find some species, so they may be sharing common characteristics. Okay, they may be sharing common, so I'm talking about these continents. Okay, so they may be sharing common characteristics. Why? Because let's say fossil records. So we do a study of the fossil. What do we mean by fossils?
The meanings of plants and animals after thousands of years.
Yes, there can be plants and animals that have, now they are buried underneath. Okay, but if we just extract rock, and you might have seen a lot of videos also that these were once living organisms. These living organisms have died and then they were submerged and over, they might have fossilified. They might have turned into rocks also. After they turned into rocks, maybe their structure is still like that. Okay, they are known as fossils. Okay, fossils. All the petroleum, natural gas that we are using nowadays, so they are fossil fuels. Okay, fossil fuels. But they are exhaustible form of resources. Exhaustible means one day they will be wiped out. All of it will be utilized. Then we will not be having any further fossil fuel. So that's why we are looking for alternate sources of energies. Okay, that's why we are building nuclear energy, wind energy. Those are renewable sources of energy. Wind energy is renewable source of energy. Solar energy, renewable source of energy. Okay, because energy will be needed for us. Okay, so fossil records help us to identify this. Okay, fossil record, are we understanding? Fossil record means like thousands of years back some organisms, individuals died, they're buried underneath. If we take them out, then like their bodily skeleton maybe can be identified with other organisms which are similar. Okay, so bodily structure or skeleton structure may be similar. So this is what I mean like helps us to identify that maybe some genetic variation has happened, evolution has happened. Okay, so long back dinosaur species were there,
Yes.
but do we have dinosaurs now? Do we have some species that have similar bodily structure, skeleton structure? Huh? Komodo dragon, huh? Lizard also. I mean, like they are kind of maybe like similarities there. Evolution has happened, but those were very big in size. Okay, so that's the thing. So a lot of these things, molecular biology, if we study the molecular structure and all, we'll be able to identify. So this is the thing. So this is all about evolution. You need to read about this particular article. Okay. Now we are going to discuss about molecular basis of inheritance. And before that, let us talk about another topic. Okay. So I think that topic will be easy and important for you to understand. This is, let's say like, here now we are going to discuss about one important thing, which is cell theory. Then we are going to move to the next topic so that like you can build the connectivity. Directly going to the topic becomes little difficult. Cell theory. So what do we mean by cell theory? So what do we mean by cell?
Basic.
Yes. Basic unit. So here we are talking about cell with reference to living beings. Okay, with reference to living beings. So here when we talk about cell, right, cell is the fundamental structural unit of life. Fundamental structural unit of life. So we may have certain, I mean like certain species that are single celled. We can have a species that are multicellular organism. Okay, single cellular also, multicellular can also happen. So basically, if we talk about cell theory, three important things are associated with that. Okay, first, the cell theory is, let's say, composed of three important things. Number one is that all living beings are made up of cells. All living beings are made up of cells. Okay. Anything which is living, they are made up of cells. Either they have single cell, they have multiple cell in the body. All right. Second, cells are the basic structural and functional unit of life. Cells are the basic structural and functional unit of life. Third, okay, we will understand about all these statements one by one. Third, okay, third, now, so this is cell theory. Write it down if possible. Okay. So we will discuss what do we mean by cell theory. So I have written 3 points associated with this, that all living beings are made up of cells, either single cell or multiple cell, right? Second is, cell is the basic structural and functional unit of life. Okay, structural means cells give structure to the body, it gives function to the body also. Okay, we'll discuss about that. Third is, cells arise from pre-existing cells. Okay, means like reproduction happens. Cells arise from pre-existing cells. So here when we talk about this particular thing, cells are the basic structural and functional unit of life. Now if we talk about human beings, so we are multicellular organism. I mean, like, we have multiple cells, and cells come together, they form tissues. Tissues come together, they form organs, and multiple organs come together to form organism. Okay, so we are a multicellular organism. We have multiple types of cells, like we have hair cell, we have bone cell, we have blood cells, like we have different types of blood cells. I mean, like RBCs, WBCs, right, platelets, etc. So like, you know, we have blood cells, different type of that. We have, I mean, like heart cells are different, nerve cells are different. So these different types of cells perform different functions in the body. Some cells provide a structure to the body, like that. Okay, so when we talk about human beings, right, so here we have multiple type of cells. In animals also they have different types of cells, right? In plants they have different types of cells. So multicellular organisms are also there, single cellular organisms are there. So cells are the basic structural and functional unit, means like cell provide structure also to the body, they provide function also to the body. Okay, then third point, it says cells arise from pre-existing cells. What do we mean by this? It means cells reproduce. Okay, new cells are formed from previous cells. Cells reproduce. How cells reproduce? When we by cell division, that is known as cell division. Okay, so we'll understand about cell division also. So basically when we talk about, so have you written down this point? Okay, so when we talk about cell division, okay, so now let us discuss about cell division. So what do we mean by cell division? Basically cell division is the process through which replenishment of cells happen. Okay, there are different types of cell division. If I talk about the human beings, okay, so I mean, so we have how many pairs of chromosomes in the body? 23 pairs of chromosomes. 23 pairs, I mean, like, you know, we have 46 chromosomes. Okay, 23 pairs. So out of those 23 pairs of chromosomes, okay, 22 pairs are of one type and one pair of other type. Okay, these pairs of chromosomes are, they consist of somatic cells. What? They consist of somatic cells. Then this particular pair, it consists of germ cells. Okay, or reproductive cells. Reproductive cells. Now when we talk about somatic cells, for example, if I talk about blood, I mean, if I talk about RBC, or if I talk about WBC, RBC means red blood cells. When we talk about WBCs are also of different types, right? You might have heard about that. So when we talk about RBCs, then RBCs, I mean, like red blood cells, they are created in the body. Okay, they are created where the red blood cells are created. Yes, so they are created in bone marrow, right? So now the thing is, like, red blood cells are created and they have a certain lifetime after which the cell will die out. Then, I mean, like, you know, similar cells will be reproduced again. So this kind of reproduction, okay, this kind of regeneration of cell, it is not giving rise to the individuals. I mean, like, you know, it is not giving rise to the individuals. It's for maintenance of life, basically. Okay, so these kind of cells, I mean, like, you know, that divide to form identical daughter cells. I mean, like, one cell divide to give rise to identical cells. They are known as somatic cells, basically. Then there are certain cells in the body that are, I mean, like, you know, that are germ cells. Germ cells means they help in the reproduction. Okay, for example, in males we have what? Huh? We have XX, XY. In female, XX. What is XX, XY?
Gender defining
Huh?
gender defining
हाँ, gender defining genes होता है. So basically now here when we talk about this particular thing, so here I mean, like, these are germ cells. When these germ cells will combine So they will give rise to a baby offspring, and here there will be a process of cell division, okay, process of cell division. Here this particular process of cell division is known as what? Its name is mitosis. Here it is known as mitosis. Mitosis means one cell will replicate, okay, will give rise to identical daughter cell. So that process of cell division is known as mitosis. It is there in 9th standard NCERT book, okay. 9th standard book, there is a chapter where they have talked about this. And then in this particular thing where cell divides, here this particular process is known as meiosis, okay, meiosis. So what happens in mitosis? One cell give rise to two identical daughter cells. Two identical daughter cells. And over here, a cell will divide into four haploid cells. These cells over here, they are known as diploid cells in case of mitosis. These cells, these cells are known as diploid cells. There is a name to it, diploid cells. Over here, these cells, diploid cells are the identical daughter cells. The cells that are formed, identical daughter cells, they are known as diploid cells. In meiosis, four cells finally will arise, okay, four cells will arise, and these cells will be different, okay, these cells will be different from the, I'm just marking it differently so that we can understand. So these cells are going to be different from the parent cell, and these cells are non-identical cells. Non-identical means they are not similar to one another. They are non-identical cells, and these cells are known as haploid cells. Haploid cells. There are multiple processes over here, okay. Mitosis, in meiosis, two stages happen, okay. So prophase, anaphase, metaphase, telophase, multiple intermediate phases are there where one particular cell will divide to finally form four daughter cells, and these daughter cells are not identical to one another, and these cells are known as haploid cells. Over here, these cells are known as diploid cells. So this is cell division. Cell division is also a part of cell biology. Cell biology means whatever function is performed by the cell, it is part of it. Cell division is part of it. Why cell division is important? Because it helps the organism to survive, okay. All the cells that die out, there will be regeneration of those cells also, okay, before they die out, okay. So I will ask a question and I will ask you to give the name of it, okay. So some people have let's say their RBC shape changes. They are facing some problem related to red blood cells and they need to go for blood transfusion. Yes, sickle cell anemia. Okay, so sickle cell anemia means generally RBCs are the cells that carry oxygen, right, in the body. So RBCs are important for the bodily function. But some people, their shape of the cell becomes it becomes sickle. Sickle shape. Its oxygen carrying capacity reduces, and then there are multiple processes, multiple ways. To kind of treat this particular person. One is very frequently he may go for blood transfusion. Blood chढ़ाना पड़ेगा. Then he'll be. Recently some genetic engineering methods have also been approved. Okay, so genetic engineering methods have also been approved. So in US and UK, so one genetic engineering method is known as. Have you heard about these names? Huh? See, blood transfusion is a regular process. His blood is getting what we say. Losing the capacity of carrying oxygen. So regularly he is changing the blood. Then there is another thing where bone marrow transplant will work. One can go for bone marrow transplant. but he needs a person who will donate the bone marrow to him, but donor's bone marrow should, his bone marrow should be compatible with the donors. And donor should be willing to do it. Okay, so a lot of complication is there. So now apart from this, yes, Shri Naiki.
Sir, what exactly is bone marrow, sir?
Okay. Basically, if you talk about the skeleton, I'll give you an example. So inside the bones, let's say I have bone. Okay, I'm just drawing a short diagram. So let's say this is bone pipe shaped, and inside these bones, right, some softer, softer things are there inside the bones. Some softer things are there. So these softer things are basically bone marrow. Okay, from here, blood cells are generated, RBCs are generated. Okay, from where RBCs are generated. So this is known as bone marrow. If you have come across, maybe let's say if you have come across any picture of it. So inside the bone, there are softer structures. Color, halka maroon color. Why I'm not naming the color, it depends which portion of the bone we are into. Okay, so they are softer inside the bone, they are softer, they are known as bone marrow. Here blood cells are generated. So bone marrow transplant can happen. It is a very costly, very complicated surgery, okay, that people do. But apart from this, genetic engineering method has been kind of approved. One particular thing is Cas-GV and Life-Genia. These are two things that have been approved. Okay, I'm just writing it. Okay, its name, maybe you can write it down. Okay, so here, from this discussion, we move to sickle cell anemia. Okay, we move to SICK. Anemia. Okay, so there are multiple ways of treatment. These are conventional treatment is blood transfusion, and a surgical process is there, bone marrow transplant, then there is another thing recently approved, okay, in USA and UK. So they have Cas-GV, GEVY, and this is another one, Life-Genia. Okay, so they are also helpful for sickle cell anemia. It is a kind of genetic engineering method, okay, which have been recently approved. So this is beneficial, for the treatment of the people. That's the thing. So why we are discussing different cell types? So that we have the understanding of it. So when we talk about, mitosis, here we have sperm cells, right, and females have egg cells, right. They come together, they form gametes, and which finally kind of a turns into a baby like that. Okay, so that happens. So it is these are genetic cells, and they carry the genetic information from the parents to the babies. Okay, so here kind of genetic information from both mother and father are taken, and so the child will be having more capacity to survive, because genetic variation will come. Okay, genetic variation is important. You might have heard about that there are multiple, let's say people from different religious group or maybe caste group who do not marry outside their own community. Okay, so for example, You might have heard about these people, like Parsis. So Parsis, I mean, like, so they do not marry outside their Parsi community. Okay. So the thing is, genetic variation has not happened much.
Become weeks.
Genetic variation has not happened much, and so now, I mean, like, much variation has not happened. So now they are more prone or susceptible to diseases. Okay. If let's say, like, one person gets affected by a disease, then same thing may be genetically passed over to the others, and they are not marrying outside. So the thing is, they will kind of, I mean, like, they are going to face a lot of difficulty in that case. So that's why, I mean, like, in religious views, I mean, like, so that's why, I mean, maybe among Hindus, they don't marry within their gotra. I mean, so a lot of things are there. I mean, like, they'll marry outside. Okay, so a lot of concepts are there. Okay, so it has some scientific basis also. Okay, that's the thing. So,
Children related issues are also there. Children's related issue, like if they got married, their children will not survive after birth. There is issue between.
मतलब it's not as immediate. I mean, like, it's not as immediate. Generally, I mean, I can understand. I mean, like, it's not as immediate. It depends. There is one case I'll talk about when we talk about bloods. Okay, blood group. I mean, like, people have different types of blood group. There is a particular combination of blood group which is not kind of, let's consider father has a certain blood group, mother has a certain blood group, and then they are not compatible with one another. Then the child will be having issues. Okay, child will be having issues and will not be able to survive much. Okay, so it happens because, let's say, like, if the mother has any negative blood group, AB negative, okay, and, like, so, and father has some maybe AB positive, I mean, which is entirely different, then there will be problem. Okay, so there is a name of a disease also, like, that the child has, Leigh syndrome, I think, Leigh, Leigh, Leigh syndrome. So we'll discuss about it. There is another thing. Okay, there is another thing which is, let's say, like, we are going to discuss about like, DNA mutations and all. Lot of things we are going to discuss. Why I discuss these things? So that, like, basic things must be little clear, so that, like, we can move further. Okay. So when we talk about molecular basis of inheritance, anything you want to write it down here? Any question? Okay.
He's saying we should ask during marriage.
So many countries, some European countries have rules like this before marriage.
Yeah, before marriage they generally take, I mean, like, medical profile.
Yes.
Okay. And so here in India,
Yes.
people taking medical profile is long far away. People do not see the face also कि हाँ वो इनके घर वालों ने उनको पसंद किया, उनके घर वालों ने इनको पसंद किया, they have seen only photo, okay. हाँ, no, it becomes okay. But the thing is, like, in foreign countries, or let's say maybe in India also, in certain multinational companies, if you have applied for a post for a job, they will ask you to get your blood tested also. I mean, like, before you join the company, okay, so आपके blood report वगैरह वो ले लेते हैं, because, I mean, like, emergency situations, okay, they take the blood group and all. So it is a good practice. I mean, like, in the beginning itself, the stage is there. I mean, like, in future, if you face any difficulty, so they'll be like, they'll be coming up with some kind of solution. Okay, solution in the sense they know कि हाँ, this is the blood group of the person, right? This is the emergency contact and all. So these things, professional things must be there. But in India, I mean, like, when people are marrying, okay, nowadays, I mean, like, entirely a lot of things are happening. Like, guys are more fearful of getting married than girls. Okay. So any case you have heard about recently? So many. हाँ.
सर कुछ साइंस का भी था ना इसके एआई रिलेटेड समथिंग
AI related?
मींस साइंटिस्ट इंजीनियर था वो एआई का तो ऐसे मैंने तो सोच
अच्छा, सुसाइड कर लिया.
yes.
Okay, a lot of things are there in India. All right. देखो, our society is kind of it's like progressing in some area, going backward in some other area.
Yes.
Simultaneously, many things are happening in this society. Okay, progressing in some area means technologically we are adopting many things. Okay, for example, in India we have huge population. Okay, we are adopting artificial intelligence. Why artificial intelligence is needed in India? It's not needed much. It's needed in those countries, European Union, I mean like those countries where labor population is less. Okay, I mean like cost of having a labor is very high. So now they try to automate the things so that cost of production of things goes down. Okay, I mean like because labor is very costly over there. In India, huge amount of people are there who are unemployed. Okay, kind of. There is no dearth of labor. Dearth means labor की कोई कमी नहीं है इंडिया में, right? Huge number of people are unemployed. Then artificial intelligence comes. People who are already employed, they are about to lose their employment because of AI. Okay, I mean like this is the thing. So technologically we are adopting the things, but otherwise we have not created avenues for these people to get employed because skill generation is not happening. I mean like people are not trained, people are not imparted with the skill. अगर skill empowerment नहीं होगा, kind of people are not going to get jobs. And then political parties are there, they're pushing people backward कि वो बजाओ. Lot of things are there. So kind of that way the society is not progressing.
Too much organic in nature.
Huh?
Indian society is too much
Inorganic in nature.
organic in nature.
Organic in nature. हाँ मतलब he has used a very sophisticated word related to this. Okay, so now let us discuss about molecular basis of inheritance. Okay, so there is another topic that we want to discuss quickly. Molecular basis of inheritance. Okay, molecular basis of inheritance. So basically when we talk about inheritance, it means acquiring the characteristics of previous generation. Okay, acquiring means like inheriting something. Okay, inheriting means like parents pass on their genetic information to their next generation. Okay, so how they pass on their genetic information to the next generation? They pass on the genetic information to the next generation through what? Genetic cells, gene cells. Okay, through genes. Okay, through gene cells. So what are gene cells? Basically we talk about DNA. What is DNA? Deoxyribonucleic acid. So we are talking about a particular type of nucleic acid. So in the nucleus, within the nucleus, we have multiple types of acid, right? Like oxyribonucleic acid, okay, or ribonucleic acid, RNA, deoxyribonucleic acid, okay, which is DNA. Now when we talk about DNA is something that carries the genetic information because DNAs come together, I mean from both parents and they form the baby basically. Here I have given the structure to you, I mean so images here, so we'll quickly analyze this image so that yes. So I can make it bigger here itself. Okay. Can we see this structure? Is it visible to you? Shinaiki? कैरी करता है. आरएनए का काम है ट्रांसलेशन. विल डिस्कस अबाउट इट. ठीक है. तो प्रोटीन सिंथेसिस में काम होता है, ठीक है. ट्रांसक्रिप्शन, ट्रांसलेशन, विल डिस्कस अबाउट दैट, ठीक है. तो बेसिकली हियर वी आर टॉकिंग अबाउट, so nucleic acid means like they are found inside the nucleus. Okay, so DNA, deoxyribonucleic acid. So DNA is made up of multiple things, as I recently discussed. So every DNA, okay, so like over here we have different types of these nucleic acids. One is cytosine, guanine, adenine, thymine. Okay, so DNA is double helical in structure. Double helical means एक इधर से एक इधर से. Okay, so DNA is like structural wise also it is more strong. Okay, one thing. Then in DNA there will be, I mean, like certain base pairs that connect with one another. ATGC. Okay, in case of RNA we have AUGC, adenine, uracil, then guanine and cytosine. Only one pair is different. It is uracil and here it is thymine. Okay, so these are nucleobases of DNA. These are nucleobases of DNA. So now if I talk about this particular thing, if I talk about this particular structure, in this particular structure we have something which is known as phosphate group. What? We have phosphate group. What does this phosphate group do? This phosphate group provides Yes, it links, basically it links, I mean, like, you know, different things together. It provides a structural support, okay, to the DNA structure over here. So phosphate group. Now I will give you a simple, let's say problem means question for you to identify. Take any of the base over here, take any of the nucleobase over here, either cytosine, guanine, adenine or thymine. Identify how many carbons are there in any one of these things. You have the chemical formula written. Identify how many carbons are there. Yes, he has made a kind of guess.
So thymine is 7.
Carbon. Huh? 5, right?
So 6 तो benzene में ही होगा, 1 thymine 6.
No, you have to identify, take any of these nucleobases, either cytosine or guanine or adenine or thymine. Take any of them, any one of them, and identify how many carbons are there. कैसे 6? किधर? Yes. Shri Naiki, would you like to make any guess?
cytosine में जो है मतलब संख्या 4 पे आ गई, cytosine में 4 ही है.
Repeat the question, sir.
1 2 3 4 NH2 लगा.
हाँ. अच्छा. Question is, in this particular structure, in any of these structure, how many, in any one of these structure, how many carbons are there? Which one did you take? Adenine 5. Okay. So one carbon will be here, other will be here, third will be here, fourth will be here, fifth will be here. 5. Kind. And so in every one of them there will be 5 in general. So over here na, you are saying कि यहाँ पे 4 होगा, right? You are saying this. क्योंकि n is here also. 1, 2, 3, 4. This structure. Cytosine. NH2 and H. 4. NH में. Cytosine, it seems कि there are 4. But generally, I mean, like, you know, when we read, so 5. 5. ठीक है, generally 5. हर किसी में 5 है, इसमें 4 है. ठीक है. Here like maybe 1, 2, 3, 4, 5. ठीक है. मतलब these are benzene structures. One carbon is replaced by a nitrogen. So that's why they are known as nitrogen base pairs. Okay, nitrogen base pairs, because nitrogen provides function, okay, to this particular benzene thing. Okay, now it is not known benzene, it is nucleobases. Okay, so basically here when we talk about this particular thing, structure of DNA, components of nucleotides. So over here, phosphate group, a molecule containing phosphorus that links nucleotides together. 5 carbon sugar in DNA. This sugar is known as deoxyribose, nitrogenous base. There are 4 types. Okay, we are talking about DNA. 4 types of nitrogenous bases are there: adenine, thymine, guanine and cytosine. Okay, base pairing. How the pairing happens? It happens in ATGC format. Adenine pairs with thymine, guanine pairs with cytosine, like that. Okay, so over here we can see, okay, although let's say here they have not mentioned that way, so they pair it that way. Now, central dogma of molecular biology. Okay, so there are certain key terms that are asked by UPSC in the prelims exam. Key terms in the sense, in the prelims questions, they may ask about the what do we mean by transcription? What do we mean by translation? I mean, like, they don't ask the definition that way, but they will give sentences. Okay, then you need to identify. Okay, so here now when we talk about this particular thing, central dogma describes the flow of genetic information within biological systems. It outlines how DNA is transcribed into RNA. Okay, so this process, of conversion of DNA into RNA, this is known as transcription. Okay, transcription. So it goes through 3 processes, 3 phases: replication, transcription, translation. Okay, so replication means one is changing in kind of copying. There is one thing, it is replicated into multiple. Okay, so replication, the process by which DNA makes a copy of itself before cell division. Transcription, it is the synthesis of messenger RNA from DNA template. Okay, so from DNA messenger RNA is formed. Then translation, it is the process by which ribosomes synthesize proteins based on sequence of mRNA. So mRNA, messenger RNA, provides information, and using which ribosomes, what they do, ribosome basically, it synthesizes the what? It synthesizes the protein. Okay, so do you know what is ribosome? Cell organelle, right? So ribosome is cell organelle. So it helps in the protein synthesis. Okay, so that's the thing. So ribosomes are located at cytoplasm. Okay, and any other additional information? Inside the Golgi apparatus. Okay.
So what exactly cytoplasm is inside cell wall?
See, cytoplasm, when we talk about cytoplasm, inside the cell, okay, let's say this is the cell. Inside the cell there is a jelly-like fluid. Okay, jelly-like fluid. Where? So inside the cell we have jelly-like substance. I mean, where cell organelles are suspended. Okay, that is known as cytoplasm. Okay, that's the thing. So this is, I mean, here we need to understand about these keywords. What keywords? I mean, what is transcription? Synthesis of messenger RNA from DNA template. Okay. Now here, if we talk about DNA replication also, DNA replication means, one DNA is copying itself into multiple ones, multiple copies of DNA. So it goes through 3 phases. One is unwinding, complementary base pairing, and formation of new strands. Okay, so it is kind of copying itself. Then when we talk about transcription, transcription means the process that we have discussed. So it goes through multiple stages internally. I mean, I have not taken those images over here. So the thing is, transcription, we need to understand what happens in transcription. From DNA, messenger RNA is formed. Okay, messenger RNA is formed. So process of transcription happens in 3 stages. One is initiation. RNA polymerase binds to a promoter region on the DNA. Then elongation, the RNA polymerase synthesizes mRNA by adding complementary RNA nucleotides. Okay, AU, GC. AU means, what is A? Adenine, uracil, and cytosine, guanine. Okay, so they kind of, these are nucleotide base pairs. Okay, so elongation happens, and finally after transcription continues, then reaches a termination stage. So these are the stages of transcription. Then what do we mean by translation? Translation.
Formation of protein with the
Help of the information provided by messenger RNA. Okay, so translation is the process by which ribosomes synthesize proteins based on mRNA sequence. Okay, based on mRNA sequence. So basically, I am directly discussing about some of these things. Some of you may be finding it difficult because NCERT book is very important. We need to read 9th standard NCERT, 10th, 11th. I have brought some NCERT books just to show. Okay.
The mRNA, can you please just explain, sir? mRNA vaccine. mRNA vaccine.
Okay, mRNA vaccine. All right. So basically, when we talk about COVID-19, that was a virus. Okay, that virus was RNA virus. What? RNA virus. Now, when we talk about viruses, we need to suppress the viruses so that viruses do not replicate itself in the body and they do not suppress the other functions of the body. So how do we tackle the viruses? There are multiple ways of addressing it. One thing is creating or making vaccines. Okay, one thing is creating or making vaccines. How vaccines help us? Vaccines actually introduce the same virus in our body. What it introduces same virus in our body in a form, either it is attenuated. Attenuated means its power or its strength has been reduced significantly. Okay, whatever is the strength or power of a real virus, there in the vaccine, similar virus strains will be there, but its power is reduced significantly. Okay, or dead virus will be there. Dead means dead virus will be there. It is injected in our body. When we inject it into the body, what does body do? In our body, we have internal defense mechanism. The WBCs, white blood cells in the body. So they identify what is foreign substance. Okay, any foreign substance that is there in the body. So it fights against it. Okay, so if we are giving something which is of very small strength, so our body will be able to develop immunity against it. Okay, it is going to develop immunity. Once the immunity has been developed, then what it is going to do, in future, if we are attacked by the same virus, the actual virus, our body has already identified that this is a foreign material and it has developed immunity, means it has strengthened itself. Then in future, any real virus, if it attacks, so it will not cause much problem to our body because our defense mechanism has already been trained. So this is the mechanism through which vaccines are functional. Okay. So when we talk about mRNA virus, okay, basically it was popular during COVID-19, so it was made, and so this was one of the this was one of the responses that we have come up with. Not in India, different countries in USA they have made it, in Russia, Sputnik, I think they have made it. So basically in India we have produced those vaccines. Okay, so those vaccines were beneficial against COVID-19. So the thing is, it was not the case that COVID-19 has affected infected everyone. Some people who came under the attack of COVID-19, if their immunity was not very strong, so they fell ill. Some people passed away also. If your immunity is strong, if you are able to if your defense mechanism is strong, you will be able to fight. But that virus had the capacity of replicating, in the sense that it transfers from one person to another. So that's why we have actually vaccinated most of the population in the country. Okay, the initial vaccination was given to, that was also a vaccination strategy. Initially they have given vaccination to 60+ years people. they are more vulnerable because like their immunity is already like very weak. So they were vaccinated first, means if they can come under attack of the virus, so they'll be able to survive. Then they have given it to children below the age of 18 years and all. Okay, at last they have thought of like adults like us, because they feel it that way. Okay, so related to mechanism of the virus mRNA, we will discuss maybe in the next class. Okay, but like I mean a lot of things are there in the virus. I mean it's a structure we need to discuss. We need to discuss about like how the virus was formed. Okay, how testing is done. Then nasal swab, they do swab testing. I mean like you might have been tested also multiple times. हाँ हाँ मेट्रो स्टेशन पे नाक से वो ठीक है. So the thing is they take the sample and in the laboratory they amplify it. Amplify in the sense like they multiply, replicate करते हैं. ठीक है. So उसको जब replicate करते हैं तो बहुत सारा हो जाता है. Then उस particular the sample that they can collect, it has DNA. So that DNA is transcribed into RNA. ठीक है, it is transcribed into RNA. Then this particular sample is tested against the virus sample कि मतलब यहाँ पे वो similar चीज़ है कि नहीं in the lab. And if the test is positive, then they मतलब report it कि हाँ ये COVID positive है. But I have been tested multiple times. Every time I came negative. But the thing is like I'll tell you one thing. Like when COVID-19 just started, I mean like that time it was only in the news. It was about to start and all. It was in the news कि हाँ China में one person has been identified कि हाँ there is a region जहाँ पे मतलब like things are spreading. हाँ Wuhan हाँ like these things is spreading. So that time we were hearing. And some of our friend, I mean like some of my friend, they told कि हाँ चांदनी चौक चलते पराठे वाली गली में. That time मैंने कहा ठीक है चलते. So we went. ठीक है. So we had paratha and all there. We came back. They also kind of came back. So on the very first day when we came back, out of 4 people, 3-4 people, 2 were sneezing and all. ठीक है. On the second day, I also started doing it. ठीक है, kind of. All of us were affected. Everybody had sneezing and kind of we lost taste also. Some fever was also there. मतलब वो taste भी चला जाता था उसमें. ठीक है. I feel कि मतलब that time itself we were affected by it. That time मतलब like after having the parathe in parathe वाली गली. ठीक है. And so we lost taste also. And after many days of that, there was a nationwide lockdown. And in lockdown, we were under fear ki, like even if we were bringing milk, we were washing the kind of from outside. मतलब उसको detergent से साफ करके then we were having it. ठीक है. So I think like I was also affected in the beginning itself. उसके बाद throughout this COVID-19, no fever, nothing like that.
सर हम लोग प्याज भी धोए हैं.
प्याज क्यों?
प्याज बोलो.
हाँ सही बात है. ठीक है. So yeah, Shri Naiki, so like you asked a question related to mRNA virus, right?
Yes, sir.
Vaccine. So we will discuss tomorrow. I'll bring the diagram also. A lot of stages are there related to that. Okay, we'll discuss about that. Okay. So with this, now let us quickly move to the next topic. Okay. So now what happens? So we have discussed about translation. Translation is the process by which ribosomes synthesize proteins based on the mRNA sequence. Okay. So there are initiation, elongation, termination stages. And when we talk about regulation of gene expression, I mean like this time Nobel Prize in Medicine, I think, it has been awarded for 2 people who have done a study on C. elegans. Okay. So what is C. elegans? Basically it's a kind of some like kind of bacteria. It's not bacteria, some other species on which they have done some study. And here in this study what they have done is they studied the post-transcriptional gene regulation. What? Post-transcriptional Gene regulation. C. elegans. So how it is important, how it is significant, why Nobel Prize has been awarded. Basically, these people have been working on it for a long time. Earlier, people felt that it does not have any impact. It does not have kind of any significance for human beings. But the thing is, later it was analyzed that, like, this particular study that they have done, post-transcriptional gene regulation, it does significant, let's say, relation with human beings also. So that's why these people have been awarded with Nobel Prize in Medicine this year, I think 2024. Okay. So it was in C. elegans. Why I'm talking about this post-transcriptional gene regulation? Because we need to understand what is gene regulation. Like in our body, we have different types of genes. And then there is a keyword which is known as gene expression. Gene expression means we have genes in our body, but these genes are controlled, they are regulated, okay, by internal mechanism. So regulation means some genes are allowed to express, some genes are not allowed to express. It happens internally in the body. The genes that overexpress, they may cause diseases or problems in our body. The genes that underexpress, I mean, like, they may also cause problems. But some of the genes, if they are expressed, it is going to cause problems. So all those gene regulation processes happens internally in the body. So now there are multiple stages at which gene regulation happens. Okay, so these are known as regulation of gene expression. So why I am connecting it with it? Because, like, it was there in the newspaper multiple times. So you need to read the Nobel Prize in Medicine. Okay, Nobel Prize in Medicine.
Micro
Micro?
RNA related to the same topic.
Yeah, no, basically, I mean, like, they have asked questions, okay, in the past, microRNA, satellite RNA. So there are different types of RNAs in the body. We'll discuss about that also, okay. So here they have talked about, okay, they have talked about regulation of gene expression, okay. So we are talking about regulation of gene expression. One is transcriptional regulation. What do we mean by transcriptional? Factors that enhance or inhibit transcription can determine how much mRNA is produced from a gene. Means, like, we know transcription from DNA, mRNA is produced. But how much mRNA is produced, right? It is regulated, right? What is the quantity of mRNA produced? It is regulated. So that is transcriptional regulation. Means how much DNA is converting into mRNA. Its regulation talks about the how many mRNA will be produced. Okay, regulation is important. Then we have post-transcriptional regulation. Means, like, mRNA has been produced, but after then the regulation that is being done, that is post-transcriptional regulation. So modification to mRNA after transcription can affect its stability and translation efficiency. Okay, means mRNA is produced, after then regulating it, okay, so that, I mean, like, after this, what is the process which is translation, okay, means like protein synthesis, okay. So modification to mRNA, like modifying this particular mRNA which is produced, right, after transcription, it can affect the stability of the mRNA also, and it can affect the translation efficiency, that how well the proteins are synthesized, okay. So that's the thing. And then we have translation regulation. Translation means after translation, proteins are made, okay, means this is the thing. So it says control mechanism can influence how efficiently ribosomes translate mRNA into proteins. That's the thing, okay. So basically it is connected. So what you need to do, like tomorrow I'll also discuss, like, I'll take a topic where we are going to discuss different types of RNAs, okay. I mean, like, in the past UPSC has asked about microsatellite RNA. They have talked about, let's say, various types of RNA. So we need to discuss about different types of RNA also. We'll discuss tomorrow, okay. So let us quickly discuss about biotechnology, okay, biotechnology. So what is biotechnology? Yes, application of technology in the field of biology. It is biotechnology, okay. Basically, like, we create processes where help of technology is taken, right, for creating biomolecules maybe, or for altering the biological systems. Or why biotechnology is important? Because it helps us to do many things. For example, if I talk about, let's say, genetically modified organisms, GMOs, or genetically modified crops, okay, so here biotechnology is used, okay, to modify the genes. Whenever we are modifying the gene, we take the help of the biotechnology, okay, I mean through biotechnology we modify the genes. So why we do have genetically modified crops?
To deal with issues like droughts, pest attacks.
So we create pest resistant crops, right, and drought resistant crops, or those crops that will be having more productivity, like that, okay. So biotechnology is important. Even when we create vaccines, creation of vaccine is also a process of biotechnology, okay. Vaccine creation is also important. Any other thing that you can share ki, this is done using biotechnology, any application of biotechnology?
Sterilizing mosquitoes.
Sterilizing mosquitoes, very technical.
Insect mosquitoes.
Hair transplant, yeah, biotechnology. GM crops, yes, genetically yes. Any example you would like to give? Any example of biotechnology?
So GM mustard.
Yes, GM mustard. It's genetically modified mustard.
इंडोस्कोपी वगैरह करते हैं, वो तो बायोटेक्नोलॉजी है।
किसमें?
जैसे कि मेडिकल फील्ड में जो इंडोस्कोपी करते हैं ना बॉडी के,
हाँ, बायोटेक्नोलॉजी है एप्लीकेशन, मतलब it does have application in let's say medical field also.
HYV seeds, sir.
High yield variety seeds, okay, green revolution, okay. So basically biotechnology does have many applications, okay. So biotechnology can be defined as the use of living organisms, systems or processes to develop products that benefit humanity. This is a broadly defined biotechnology, okay. So when we talk about principles of biotechnology, so biotechnology is founded on core principles that guide its application. One is genetic engineering. We'll discuss about genetic engineering. What do we mean by genetic engineering? When we are making changes in the genes, altering the genes, either we are taking out some portion of the gene and throwing it away, means deletion of addition or modification. So any changes in the genes that we do, right, that is this particular process involves genetic engineering. So genetic engineering involves manipulating an organism's DNA to alter its characteristics. This can include adding new genes, deleting existing ones, or modifying gene expression. I'll talk about one example. You might have heard about a keyword which is designer babies. Designer babies. Have we heard about it?
Yes, sir.
Huh?
Yes.
So what is designer baby?
Parents give the traits which they want in their baby, and doctors or labs modify or also provide some sperm or eggs respond to that. In test tube babies में ऐसा होता है.
हाँ, मतलब like we might have heard about assisted reproductive technologies.
Yes.
What? Assisted reproductive technologies, ARTs, or test tube babies and all. So basically what happens, I mean before zygote is created, what they do, they alter the genes of the baby initially. Doctors do it. Why they do it? So that they can impart certain desirable traits in those babies. Okay, maybe someone wants that my baby should be running as fast as Usain Bolt. Okay, maybe someone wants that my baby should have blue colored eye. Maybe someone wants that my baby, I mean some enhanced characteristics. Okay, so that is designer babies. It can be done using genetic engineering, but this is ethically not good. Okay, why? Because lot of things will happen. I mean so like if something goes wrong during the process, that baby may have altered, I mean, like, you know, maybe mental faculty, or many other things can also be there. And then if designer babies become popular, then naturally born babies, they will feel inferior. That's also a thing, na? Means like,
Everybody can't afford
huh?
Everybody can't afford that.
Everybody can't afford it. I mean, like only wealthy people, people having crores of rupees, they can afford this particular thing. So here genetic engineering. So here what we are doing, we can manipulate an organism's DNA, right? So designer babies is an example. Then we have genetic modified crop also. So there also genetic engineering happens. Application, it is used in creating genetically modified organisms, developing gene therapies, and producing recombinant proteins such as insulin. Okay, so gene therapies, I discussed about 2 gene therapies today. One is Cas-GV and Life-Genia. So they are effective against Cas-GV and Life-Genia. They are effective against
Sickle cell anemia.
sickle cell anemia. Okay, we have discussed it with reference to sickle cell anemia. So they are gene therapies. Okay, so I'm just writing it one again, Cas-GV and Life-Genia. Okay, so they are kind of I'll tap here. All right. So this is the thing. So genetic engineering is understood. Like broadly we are understanding. Then bioprocess engineering. Okay, bioprocess engineering. So what do we mean by bioprocess engineering? It focuses on designing and optimizing processes that use biological systems to produce valuable products. Okay, valuable products using biological systems. For example, if I talk about biofuels, if I talk about pharmaceuticals, I mean, like, you know, tablets, etc. and all, food products. Okay, so here like, you know, bioprocess engineering may be done. One of them is known as fermentation technology. Okay, then we have cell culture technique also. Cell culture, I mean, like growing a large number of cells under controlled conditions for research or production purposes. Okay, so these are, then we have molecular biology techniques. Okay, molecular biology techniques. So these are different kind of techniques. So techniques such as polymerase chain reaction, PCR, RT-PCR, like we have heard during COVID-19, RT-PCR hota tha test. RT-PCR, dengue में तो सुना नहीं मैंने. I've heard related to COVID-19, RT-PCR test. That test was named as RT-PCR test. Reverse transcriptase polymerase chain reaction. Then DNA sequencing. Cloning are essential for manipulating genetic material, studying biological processes. Then bioinformatics is there. What is bioinformatics? Combining biology with computer science to analyze biological data, particularly genetic sequences. It plays crucial role in genomics and proteomics. For example, I have given you an example today where people have created artificial intelligence systems, okay, that helps in identifying protein structure.
Yes.
Can anyone tell me its name if you are aware with the current affair? AlphaFold 2. Okay, so AlphaFold. So it was developed by Google. Okay, I mean, like, you know, Google's DeepMind. So one vertical of Google is Google DeepMind, where they are working on artificial intelligence. They have created this system where if we put any kind of name of protein, it is going to tell us the structure also. And this will help us to create newer types of protein, and like protein synthesis and all. So it is actually helping huge number of people, right, globally for this. So we'll see also, like, you know, we'll see also AlphaFold. Let's see if internet is connected. AlphaFold 2. This is AlphaFold 2 protein structure database. Okay, so this is the artificial intelligence system developed by Google DeepMind, like that. Okay, so they have given many examples also. E. coli, we have heard about E. coli multiple times. Gut bacteria, I think. E. coli. Showing results of E. coli. Okay. So if we have to see the structure, go to Uni Pilot. Let's see what is. Okay, so this is the basically database where if we put the name of anything, it shows the structure also. It talks about, let's say, like it provides all the information related to that. This is the structure of E. coli.
So the bacteria, no, sir?
Yes, yes, gut bacteria. I mean, like, you know, it's. It's created by artificial intelligence system. Okay, so prediction of protein structure used to be one of the most toughest work earlier. Okay, but they have created the AI system. If you have the protein sequence, put it there. If you know the common name of the protein, you put it there and you will be able to visualize the protein structure. And since people now can visualize the protein structure, they can, I mean, like use it to synthesize new proteins also. Okay, so I mean, like many things can be done over here. Like we are not doctors, so we understand little less about the protein structure and all. Okay, so this is the thing. Okay, so we can download the file and all. So Google, like Google's DeepMind, they have solved this problem not for once. I mean, like for the humanity. Okay, so they have using this particular system, billions of protein structures have already been identified. Okay, they have already been identified, and this database is now becoming better day by day. Okay, so this is the thing. All right. So we were discussing about bioinformatics. So this is bioinformatics, means like biology with information technology. So they have kind of connected it and they have created AI system. And the person, there were many people who worked on it. One person was a physicist and then he was a chess player also. So they have utilized their knowledge in certain combination. Now, let's say it has benefited the humanity. How? It is bioinformatics. I mean, like, you know, so retina, they capture the information. So now the main thing, what one important aspect is there, that our fingerprint also changes over a period of time. It evolves. Okay. So it changes. Our fingerprint does not remain the same whole life. I mean, like a lot of things happen. Okay, so they use it, but like anyhow the database is so good that like they are able to capture and identify individuals with this. Okay, now think about this particular thing. Although let's say like there are limitations to the government that they cannot use our biometric data anywhere and everywhere. Okay, because of privacy issues. But the thing is, long back what used to happen, that people who are there in cities, metropolitan cities, let's say like you are traveling on bus and all of a sudden maybe a person who passes away, dies, they don't have any ID card with them. How they are going to identify who is this person, who is this deceased person? But the thing is, now since Aadhaar data is there, maybe, I mean, like they may take that kind of fingerprint thing, they may take and put it there, and like his Aadhaar information will come up.
Yes.
I mean, like this can be one of the application, but very extreme case. So I thought of discussing with you. Okay. So now let us talk about another thing. What can be the applications of biotechnology? All right. So recombinant DNA technology. So we might have discussed about it. Okay. In the past, when I had taken the kind of that classes means MGP classes. Huh? MIGP. So MIGP, like there we discussed in detail about many advanced things. Okay, here I want to discuss about some kind of fundamental things, because questions revolve around these. Okay, in the prelims exam. Now, genetic engineering, we already understand what is genetic engineering. So there are multiple techniques. One of the technique is recombinant DNA technology. Okay, inserting desired genes into host organism using cloning vectors and all. Okay, so bioprocess engineering we have already discussed. Fermentation, cloning, applications of biotechnology. Yeah. So we will quickly see some applications of biotechnology. Okay, here you need to kind of give a specific attention. So what can be the applications of biotechnology? One is biopharmaceuticals. What is biopharmaceuticals? Production of life-saving drugs such as insulin, monoclonal antibodies, vaccines. Okay, it is biopharmaceuticals. Production of vaccines, monoclonal antibodies, etc. Okay. Then we have gene therapy also. We just discussed about gene therapy, right? So gene therapy, I mean correcting defective genes to treat genetic disorders. There can be multiple genetic disorder. One of the genetic disorder that I discussed is Leigh syndrome. It's a genetic disorder. Okay.
अल्जाइमर को भी जेनेटिक डिसऑर्डर कैसे?
किसको?
अल्जाइमर को?
अल्जाइमर. अल्जाइमर जो ओल्ड एज में हो जाता है? नहीं नहीं नहीं, इट्स नॉट जेनेटिक. हाँ, इट्स नॉट जेनेटिक. हाँ, मतलब जेनेटिकली मे बी पास्ड ऑन. जनरली होता है क्या ना, विद एज, मतलब लाइक आवर कैपेसिटी टू रिटेन इंफॉर्मेशन गोज डाउन. पार्किंसंस डिसीज भी होता है, ठीक है, काइंड ऑफ. ओके, सो आवर कैपेसिटी टू रिटेन इंफॉर्मेशन रिड्यूसेस. We tend to forget the information. So that's Parkinson's disease. So it happens with age. Okay, that's the thing. So that's why it is recommended that all of you have those, kya bolte hain, good nuts, etc. Right? So it keeps your brain sharp. So there is a food that looks like brain. Kya bolte hai usko? हाँ, अखरोट. अखरोट, हाँ, walnut. So I have heard कि हाँ, walnut is very helpful for brain thing. Okay, so what is Leigh syndrome? Let us discuss. Basically, there is a defect. That defect may happen in the kind of powerhouse of the cell. What is the name of the powerhouse of the cell? Mitochondria. Mitochondrial defect होता है, ठीक है, मतलब like so inside the cell there is a component that provides energy to the cell, that is known as mitochondria. So what happens, I mean like it is there in all the cells. Okay, so if we talk about the egg cells means in case of female the kind of reproductive cell is egg cell. So in egg cell, if let's say mitochondria is defective, right, and if let's say like a zygote is formed and a baby happens, and that baby will be having defective mitochondria in his cells, which is genetically transmitted. It is genetically transmitted by the parents to the children, and that children will be having, let's say like lifelong disease. I mean like his mitochondria is defective. So like this adequate amount of energy will not be generated in the cell. If the adequate amount of energy is not generated in the cell, what will happen? I mean like that baby will not be able to do, let's say like any good activity and all, physical activity. So that's why it is kind of it is recommended that like this is avoided. How to avoid? So there was a technology, I mean like using the genetic engineering. So they have come up with a solution for it, and this solution is known as 3-parent baby in generic terms. And so it is also known as pro-nuclear transfer technology. Okay, pro-nuclear transfer technology. So what happens in 3-parent babies? It's that, okay, we'll quickly discuss about that also. So the baby, I mean like if he has defective mitochondria, so that baby will be facing problem which is I think known as Leigh syndrome. Okay, but the thing is, when we talk about 3-parent baby, what happens here is that, let's consider I'm just making a simple looking cell over here, okay, simple looking cell, in the sense this is, I have made it like a round circular, but like it's elongated, so it is, let's say, in case of male, in case of female, we need to consider, okay. So over here in this particular cell, what we have defective mitochondria. The yellow color, it shows mitochondria, let's say, but this is defective, okay. What this is defective. So if this defective mitochondria is passed on to the kind of next generation, so that baby will also have the defective mitochondria in his cell, so it is going to create problems. So how do we avoid this particular thing, like how do we avoid, or let's say like what solution we can have is that over here, this is, I mean, like so this is the female, I mean, like she wants to have the baby kind of with this person, but the thing is like her mitochondria is defective. So in that case, here we need a donor. What we need a donor. So who is going to be the donor? Donor is going to be another female who is willing to donate his kind of egg cells. Now let's consider, so this is the donor, it's the female, and so I'm just making it. Let's consider she has donated the cell, and in this particular cell what happens, I mean, like over here we have proper mitochondria. Proper mitochondria means like healthy mitochondria. So these are healthy mitochondria. Mitochondria is the powerhouse of the cell. It generates energy for the cell, okay. So this is the donor cell. So what we will do is like we'll take this donor cell. From this donor cell we'll take away the nucleus. We will discard the nucleus. We will discard the nucleus. Why we are discarding the nucleus? Because inside the nucleus there is genetic information. Like we don't want to pass on the genetic information. We just want to give the healthy mitochondria to the parent. Now from this particular cell, okay, from this cell, let's consider, let me just color it differently, maybe blue color. So what we are doing, this particular thing, which is, let's say, like nucleus. This particular nucleus is transferred into this cell. I mean, like from this cell, if we discard the nucleus, so this cell will become like this. This cell will become like this, okay, without the nucleus. So this is a cell, but no nucleus is there. But what we have done, we have taken the nucleus from this particular cell, the kind of intended mother cell, and we have imparted over here. So now what we have over here, we have a cell that has healthy mitochondria. It has the nucleus from the mother, okay, nucleus, like this is nucleus from the mother is given to this particular cell. Now this cell is good enough, okay. So if we kind of use this cell and this cell to create a zygote, okay, so now like this cell has healthy mitochondria, it has the nucleus of the intended mother, nucleus of the mother who actually wants to have baby, and it has the sperm of the father, okay. So they are going to come together and then it is going to form a baby, and this baby will not be having let's say like kind of defective mitochondria, okay. So this is a way to avoid Leigh syndrome. This is a way to avoid the genetic disease in the body, baby, okay. So it has been generally observed that mitochondria sometimes may be defective of the mother only. Mitochondria may be defective of the mother only, ऐसा Matlab, so far whatever I have studied, so in those studies they have kind of, I have come across only those articles where mitochondria was defective of the mother. So a baby is there, born, okay, a baby is born, but this baby has information from three parents. Information means not the genetic information only from two parents. One is the female mother and the male, okay. From the donor it has the cell with healthy mitochondria. From the mother it has the nucleus only, because inside the nucleus we have the genetic information. Okay, inside the nucleus we have DNA, right? So that's the thing. So this particular thing is known as three-parent baby. All right. So I hope it is understood, the three-parent baby concept also. All right. So this is the thing. Applications of biotechnology. So we were discussing about this. So pharmaceuticals, biopharmaceuticals we understood, gene therapy we understood. Okay. Then diagnostics, development of PCR based test for detecting diseases like COVID-19. So it's diagnostic. Diagnostic means identifying whether a person has disease or not, whether a person is affected by a certain disease or not. Then stem cell therapy, okay, regeneration damaged tissues using stem cells, okay. So stem cell therapy is also there. Then we have genetically modified crops, okay. So like we have pest resistant crops, drought tolerant and high yield variety crops, okay, biofertilizers, plant tissue culture. So these are basically various applications. So it finds application in the field of agriculture, it finds application in the field of environmental science also, where bioremediation. What is bioremediation? We might have read about it in NCERT books.
सर क्लीनिंग जैसे सर ऑयल स्पिल हो जाता है तो उस सेंस में तो उससे बैक्टीरिया या फंजाई से साफ करके क्लीन हो.
हाँ, राइट. बायोरेमिडिएशन मीन्स लाइक जो भी बायोलॉजिकल वेस्ट है उसको हम क्लीन करते हैं, ठीक है. इंसिनरेशन बहुत सारे वो होते हैं काइंड ऑफ सो इट इज. हाँ हाँ, क्या ऑयल स्पिल. ओके, हाँ हाँ, स्यूडोमोनास. हाँ, मतलब काइंड ऑफ अ बैक्टीरिया that is kind of beneficial against it. And because of oil spill, marine life gets endangered. I mean, like so it is not able to survive. So recently, one pond or river was in news where I mean, like oil spill has affected, and then huge number of migratory birds that were coming. Okay, so many of them were dying, and next time the kind of migratory birds came very less over there. So it was in news. So you need to remember those places also. Like, हाँ, शायद मतलब some lake was in news related to that. Okay. Then biofuels, production of renewable energy sources like ethanol or biodiesel from biomass. Biodegradable plastics, okay, eco-friendly alternatives to conventional plastics. So environmental science में भी इसका application है, and it has application in industrial applications also, like industrial processes. So enzyme for food processing, okay, microbial fermentation for producing alcohols and organic acids, biopolymers used in packaging materials. So like it does find, biotechnology finds application in various fields, okay, it does find application in various fields. So we have discussed about cell division, I mean like in the past. Today itself we discussed about cell division. So we have discussed that cell division means like replication of the cells, okay. So cell division may happen for somatic cells where one cell gives rise to two identical daughter cells, okay, and that process of cell division is known as mitosis, and the daughter cells are known as diploid cells, okay. And cell division also happens in case of germ cells, I mean like so germ cells में जो cell division होता है उसको बोलते हैं meiosis, and so it gives rise to 4 haploid cells, okay, and all these cells are different from one another, they are non-identical cells, all right. So cell cycle and cell division is a topic. This is the same topic, okay. So here, I mean, like, we are going to go into the intricacies of it. I mean, like, we are going to understand about some internal processes over it, okay. Cell cycle. Cell cycle is a tightly regulated sequence of events that prepares a cell for division. It consists of two primary phases: interphase, growth and DNA replication, and the mitotic phase, cell division. In eukaryotes, cell cycle is divided into 4 stages. So eukaryotic cell, prokaryotic cell. Okay, so basically there are 2 different types of cells. One is eukaryotic, prokaryotic. Prokaryotic means they are primitive cells. Primitive cells matlab they do not have well defined nucleus. Single cellular organisms are prokaryotic type. For example, if I talk about amoeba single cellular organism hai. So how they divide it, I mean, like, so a well defined nucleus nahi hai, they don't have well defined cell organelles also. So prokaryotics are primitive cells, eukaryotics are kind of, well evolved cells. So generally most of the eukaryotic cells are multicellular cellular organism, but single cellular eukaryotic cells can also be there. Single cellular eukaryotic cells can also be there. In this particular today's material that is there, so we have included that, okay. So now here when we talk about cell cycle means how the cell grows, how it replicates, right, how the cell division happens. So all of this is cell cycle. I mean, like, cell generation of the cell, like after cell division, cell generation happens, how it grows, how it replicates, all these total processes, cell division. So cell division has multiple phases. Let me show you the diagram first, then we will understand about this, okay. So now what happens, there is a phase which is known as G naught phase, okay, G0 or G naught, you can say it that way, G naught phase. So what happens, G naught phase, I am like, here the cell performs specialized functions without dividing. Means all cells in living organism perform certain functions, okay. I mean, like, they perform certain functions or they give a structure to the body. For example, if I talk about nerve cell, what is the name of the nerve cell? Neuron. Neuron is a nerve cell. I mean, like, neuron performs certain specialized function in the body. It transmits signal, right? Neuron is the nerve cell. It transmits signal from one part of the cell to other part of the cell. Neurons can be very small also, they can be very lengthy as well, okay, like that. So here the cell, when it is performing its normal function without division, before division, that is known as G0 phase. I mean, like, now cell is like, cell has been generated and this cell is performing its own function. I mean, like, during its lifetime. So that is, cell performs a specialized function without dividing. So this particular phase is known as resting phase or G0 phase, okay. Means like, we are talking with reference to how the cell divides, okay, resting phase. Then so here if we talk about cell cycle, here there is a phase which is known as G1 phase, and during this phase cell grows and performs normal function. I mean, like, cell is growing and performing normal function. So cell growth stage, from here to here it is cell growth stage. After then what happens, like, DNA synthesis happens, DNA synthesis, creation of DNA. So DNA replication occurs, this is known as S phase, okay. DNA replication of the cell happens, it is S phase. And after the S phase there is another phase which is G2 phase, from G1 to S to G2, and cell prepares for mitosis. So this is the stage where cell will divide, okay, prepares for mitosis. Mitosis means one cell will give rise to 2 identical daughter cells. Identical means they will be similar to the parent cell, that's the thing, okay. So in G2 phase the cell prepares for mitosis, for preparation for mitosis. After then, I mean, like, the cell division will happen, and the cell division process, the different stages associated with the division of the cell. Division means different stages associated with the reproduction of the cell. Why I'm not using the word reproduction? Because it will connote that I'm talking about meiosis, right? So cell division. So cell division, this particular entire phase is known as M phase. What? It is known as M phase. Okay. So during M phase, there are 4 intermediate phases. Okay, 4 intermediate phases. So these 4 phases are known as intermediate phases. It is prophase, metaphase, anaphase, telophase. Okay, prophase, metaphase, anaphase, telophase. So here inside the diagram, if you see, prophase means all these cells are organized in a way. In the metaphase, these cells, I mean, like, attach with one another, kind of spindle formation happens. Then anaphase is there, telophase is there. So these are internal processes in the cell division, and so this entire process is known as mitosis. So cell division happens. After the mitosis happens, cytokinesis happens. Cytokinesis means the final breakup of the cell. Final breakup means one cell was there, internal processes has happened, now they are mature enough, then they are dividing. Okay, that is cytokinesis. So now here, if we talk about prophase, so chromosomes condense and spindle forms. In the prophase, what happens? Chromosomes condense and spindle forms. After then, in metaphase, what happens? Chromosomes align at the cell equator. Okay, it aligns at the cell equator. Anaphase, sister chromatids separate and move to opposite poles. Then telophase, chromosomes decondense, nuclear envelopes reform. And then we have cytokinesis. Cytoplasm divides, forming 2 daughter cells. So these are stages. Generally, these are 4 stages, and this is the final stage, which is cytokinesis. So this entire phase is known as M phase. So we have Z0 phase, G1 phase, S phase, G2 phase. Okay. And all these 3 phases, which is G1 phase, S phase, G2 phase, they are known as interphase. It is known as interphase. And this is known as M phase. M phase means where mitosis happens. Okay, mitosis happens. So this is cell cycle. Okay, this is cell cycle. Now here, if we talk about S phase, so details of all these phases are mentioned. S phase means like this particular phase, DNA replication resulting in 2 identical chromatids per chromosome. G2 phase, this particular phase, final preparation for mitosis including organelle duplication. Then M phase is this entire phase, mitosis, nuclear division, and cytokinesis, cytoplasmic division. Okay, kind of these stages are there. Okay, so this is known as cell cycle and cell division. Okay, and after this, phases of cell cycle. So all these phases, G1 phase, S phase, G2 phase, what is this? We have, let's say, in detail I have mentioned all of it. In your free time, what I expect is read this particular thing, okay, once more, because these terms may be new for people who are not from biology. I mean, like kind of, who have not studied biology maybe after 10th and all. So most of us might have forgotten who have not studied biology after schooling, right? Yes. Anyone who is from biology field here? Anyone who has done biotechnology or anyone doctor? Shushri Naiki? Am I audible? Yes. So what was your field of graduation?
Business information, sir.
Business innovation.
Business information, medium.
Business information. All right.
Yes, sir.
Okay.
Not by
Or commerce se connected. All right.
Yes.
All right. And you?
Civil engineering.
Civil engineering. घर वर बना देंगे वो. Yes. And you? BA. ठीक है. BA. आप? BCom. Humanities. And? Master in chemistry. Okay, one person is there from science. I mean, like he's also from science. Okay. So, what's your name? Huh? Gautam. Which state you are from? Manipur. All right. Okay. Manipur. I mean, like it's good that you are in Delhi. Like, do we hear more than what is there at ground level, or we hear like what is there actually, or we hear less?
The report, ground level report, and here it is contrast.
Okay, contrasting. Okay, they're writing something else, and then what is there? Okay. Means like we have come across so many news articles and all lot of things happening. I mean like
Yes, sir.
things has happened. It's not the case that not happened. But is it better now?
कॉन्ट्रास्ट मतलब कुछ न्यूज़ कुछ पर कह रही है कुछ न्यूज़ कुछ और कह रही है
हाँ
आपस में मिडिल ग्राउंड की चिंता नहीं है
हाँ
बड़े कॉन्ट्रास्टिंग न्यूज़ है दोनों साइड
हाँ like that.
Earlier, sir, it was against the government, and at that time, at the incident, at first it broke out. The first before the incident, there was some tussle between the government and the communities, and especially the cookies. But on the 3rd of May, suddenly the class came within mutation cookies. But now, after the resignation of the minister and the coming out of President rule, then the issue subsided, and it is now within the cookie is a umbrella term. There are so many communities within them. So among themselves now they are class between them.
Okay. Cookies and meaties, like we have heard about it, right? So, which one is majority there? Majority means like dominant.
Sir, we cannot say which one is majority right now because the census is
Old.
and
Yeah.
there are so many communities shifting sites, and the border is not so rigid. There is one can pass from one side and that side, and there is the more than 100 kilometer border is porous,
Okay, porous border is there.
and so we cannot say that which one is. And mainly, metals are resided in the valley, which is only 9% of the total geographical area, and it's not more than 90% are hills, and they are resided by the
Cookies.
Cookies and Nagas.
Cookies and Nagas.
Sir, it is especially arises due to there was a lack of awareness about, especially about the forest. It is incident related to forest.
Okay. I just wanted to know that when it started, what was that thing that sparked this thing? I mean, like, I think 3rd of May. Like, why it started?
Sir, due to the there is due to the under the government of NBNC, sir, there is some how to say eviction of those who have encroached the forest area.
Okay.
Protected forest area. Earlier, there is no such rigid law, or I think there is no awareness among the tribal people, so they have settled there for a long time. So suddenly, if we say that this is not your land, then there is some misunderstanding between the government and the
And the people. Yeah.
So before May, it started from March. From March, there is tussle between the forest department and the Cookies tribe. And before 3rd May also, one of my brother who was a district forest officer in Churachampur, his office was burned down before 3rd of May.
Okay.
And so after that, the class began by saying it is not such. They say that their gate is being burned or something like a sanitary gate. It is not such things, but it is mainly the land.
All right. Yeah. So we understand a bit. I mean, like, since we are seeing it from outside, I mean, like from Delhi, so we hear only what is there in the news. So we cannot make up like what is exactly there, right? So we know like very less about it, but we know human rights violation has happened. Huge number of people have been killed. Atrocities have been done against women.
Mainly historical things also, sir, because those legislators from the state, they have passed some
Village cannot buy any land from hills area.
Oh.
But tribes can buy, and even in the village, if migrants sell their land to the tribe, we cannot buy back from the tribe without any authorization letter from the DC concerned.
Okay. I mean, like, so, like in India, what is happening? I mean, laws that are being made. I mean, like, I don't want to pass on any political statement, but what I believe, the laws that are being made. So, many of these laws are being made, I mean, like, to kind of, benefit a certain group of people at the cost of others. Many laws are there. I mean, like, many laws. Even uh, I recently heard about the UCC, Uniform Civil Code in kind of Uttarakhand. But like, I have read an article related to that. So in that article, it was clearly mentioned that the state government of Uttarakhand is not authorized enough to make a law related to Uniform Civil Code. Why it is the case? Because Uniform Civil Code is a matter of, like, it is mentioned in the directive principles of state policy, right? And this particular thing should be implemented by the central government. It should be implemented by the central government, not by the state government. Okay. And state government does not have authority to legislate on that particular matter. They have made a legislation also. And this particular legislation is so much, I mean, like, kind of, it is so much punitive means, like, if intercaste people, interreligious people, if they are there in the state, if they are cohabiting, they have to report it to the registrar of marriage. I mean, like, there is a registrar. I mean, like, they have to report it to the registrar in advance. Okay, in advance, and a person has to disclose his previous relationships also. 1, 2, 3, many are, like, people generally come across 2, 3 relationships in life. I don't know, it varies, right? So people have to disclose about that also, and that particular registrar will decide, I mean, like, on many fronts. I mean, like, so he's going to inform the parents of both the parties. Okay, I mean, a lot of things, and there is apprehension among people that the moment someone goes and discloses the information to the registrar, then that information, I mean, like, where name, father name, address, everything is given, that information may be shared by to, let's say, like, these vigilantes. You might be understanding, people who are kind of, कि बजरंग दल, lot of things. So
ये तो कोर्ट मैरिज में भी प्रॉब्लम करती है कई बार, मतलब लोग जैसे
हाँ,
इंटर वाले जैसे वहाँ पे 15 दिन के लिए
हाँ,
डिस्ट्रिक्ट मजिस्ट्रेट के बाहर लिस्ट लगी रहती है,
हाँ,
जो
हाँ,
इंटर मैरिज स्पेशल मैरिज एक्ट के
हाँ,
तहत करना चाहते हैं,
हाँ, हाँ,
तो उसमें भी दिक्कत करते हैं। ये मध्य प्रदेश में भी ये कोर्ट में एक मामला है, कोर्ट में ही पीट दिए थे आदमी को,
हाँ.
वो इंटर रिलीजियस मैरिज करना चाहते हैं।
So it's matlab, like, a lot of things I have read about it. So like, after reading, I have got an impression that this is so much anti, what we say, kind of, anti like individualistic freedom.
Yes.
Kind of. Okay.
Some couple already filed case in University of Uttarakhand.
Huh. So it is that way. Okay. So here, like, back to the discussion here. So it is phases of the cell cycle. So like we discussed about interphase, G1 phase, S phase, G2 phase, then mitotic phase, okay, which is M phase. M phase, during this phase, I mean, like, prophase, metaphase, anaphase, telophase, I mean, like, internal processes that happens, and this leads to the cell division. Okay, so this is mitosis. So regulation of cell cycle, how cell cycles are regulated. So related to that, some checkpoints are there. G1 checkpoint determines if condition favor division. Damaged DNA triggers repair or like apoptosis. Then G2 checkpoints. I mean, like, checkpoints are there. So much check and balances are there within the cell division mechanism, within our, let's say, like, bodily functions. So we don't have to think about these things. Automatically things keep on happening when the cell division will happen. So kind of it's a like, algorithmic things that happen, like, in biological systems. Recently, I mean, like, why I'm talking about this, recently you might have come across a video. NVIDIA, have you heard about this company, NVIDIA? So this is a company that manufactures hardware for computers, like so they create GPUs, CPUs. I mean, like the most of the computer. हाँ, like if I talk about, like if you have a computer, so in that you may be having a processor of Intel, kind of. So these actual hardwares are made by that company. Okay, so they are very popular for GPU making. GPU means graphic processing unit. So graphic processing unit is similar to CPU, which is central processing unit. So in computers we have CPU. GPUs are used for gaming laptops. Like if you are doing gaming and all, in those computers you have GPU systems. Okay, and GPUs are used in artificial intelligence systems also. So that particular company is a $1 trillion company. I mean, like very high valued company, right? This particular company is at par with Microsoft, Google and all, maybe much higher than these companies also. So what this particular company, the CEO of this company has done is recently he has announced a robot. Robot means, like he has had a conference. In that conference he has they have built a robot. That robot is autonomous. Autonomous means it is able to move, it is able to speak, or it's not human like, it is very small. I mean, like small in size, kind of बतख जैसे होता है ना उस size का है. It walks like that. So it is an autonomous robot that they have developed. In that robot they have put 2 NVIDIA GPUs. Okay, and large language model is also there. So this robot, GPU, yeah, GPU is graphic processing unit. Graphic processing unit. It is similar to CPU. CPU means like the core architecture of a computer that gives the compute capacity. GPU also gives the compute capacity, so it is able to process graphics. Graphics means like high level of data. And GPUs are used for artificial intelligence also. So when I saw that particular robot, I mean, like on a video, so when I saw that robot, I felt that this robot resembles a kind of, I mean, like batak kind of thing or hen kind of thing. So it resembles that thing. So we already are bestowed with, we already are blessed with huge number of, let's say, like like natural living beings. We already have huge number of natural living beings. What we are doing, humanities, what we are doing is we are destroying all those natural beings, right, for the benefit of some individuals, some corporations, and then they are creating a replica of it. Replica means, like essentially that GPU or that robot will not be having as much capacity as a kind of hen may be having, kind of. But like they are popularizing it so much. I mean, like it has created a dystopian future. I mean, like so we are destroying the natural living beings, right, like just for the benefit, right, of some individuals, some corporations and all. That's the thing. This I felt, I mean, like after seeing that. So we are going to discuss on artificial intelligence also, I mean, like later, maybe maybe tomorrow or day after tomorrow in detail. Okay, so this is the kind of cell division process. You can go through this article in your free time. And related to human reproduction, so I have included 2 articles. In one article we discussed about the various important things associated with human reproduction, and then we have included another article where we discussed about reproductive health. Okay, reproductive health, because reproductive health is also very important. So I would like you to read it yourself. Okay, although let's say like I have included the details over here. Okay, so just give a single reading to it, you will be able to understand about it. Stages of human reproduction, I mean, like pre-fertilization stage, spermatogenesis, oogenesis, ovulation. I mean, like these are the stages. Okay, and then fertilization. Okay, so like everything is there. So you just need to give a single reading to this human reproductive systems. Okay, human reproduction. Then we have reproductive health. Okay, reproductive health. This is also a very important article. So here we have discussed about what is reproductive health, what is the importance of family planning and all. Okay, so family planning involves contraceptive methods, education, access to kind of family planning services. A lot of things are there, and then let's say related to sexual health also, education on safe practices, prevention of STIs. STIs means sexually transmitted infections. Okay, kind of. Okay, then maternal health. Maternal health means health of the mother, intended mother who wants to kind of have a baby and all. So prenatal care, I mean, like during pregnancy or before pregnancy, like how she is cared upon, okay, whether she has proper body mass index, or whether she is taking the kind of proper nutrition timely or not, safe delivery. Then postnatal care. Postnatal means after the baby has been delivered, kind of how the mother is now, how her health is taken care of. Okay, so reproductive health is also a very important kind of thing. So it includes multiple things: physical well-being, mental well-being, and social well-being. I mean, like the intended mother and father, so they should be physically, mentally, and socially, they should be kind of like healthy, okay, in all terms. Okay, public health impact. So Government of India does have many projects. So you just need to give single reading to these two articles, you will be able to understand it. All right. So I mean, like there are some technicalities over here. Just read it. Maybe tomorrow we can discuss about that. Okay. Then we have another topic that we are going to discuss now. It is biological classification. Biological classification means, in general, we know plants, animals, right? So this is a two classification system. Then there are multiple ways of classifying it. So some people have kind of given classification models. Okay, so this kind of classification model is known as taxonomy. Okay, it is known as taxonomy. So biological classification, it is also known as taxonomy. It is the scientific process of categorizing and naming living organisms based on shared characteristics and evolutionary relationships. Okay, so this is the thing. So when we talk about history of biological classification, some people have been very kind of influential in it. Aristotle has been there. We know Aristotle is a philosopher, right? Like he has said something long back. Okay, Aristotle, he is also known as father of biology. I mean, like long back he kind of tried to classify the things based on observable characteristics and habitats. I mean, like whether the living being is living on the earth, or it is in water, or it is in air in the sense, I mean, like birds maybe, so that way. Then these two people are very important. Carl Linnaeus, okay, so it is he is a Swedish botanist, and he developed a hierarchical system of classification known as Linnaeus taxonomy. So I have asked a question related to Linnaeus taxonomy in one of the test papers in mains. Okay, why? Because it was in news. Okay, Linnaeus taxonomy was in news. All right. Then Charles Darwin, we know, I mean, like he talked about theory of natural selection and all. So they have played some role into the thing. When we talk about biological classification, okay, what principles are followed? Okay, biological classification is based on multiple principles. One of the principle is hierarchical structure. What do we mean by hierarchical structure? It means, I mean, like, for example, if I talk about plants, so plants include different types of plants. I mean, like different types of plants. If we talk about animals, different types of animals. So within those animals, within those plants, there can be sub-classification also. Okay. So this is known as hierarchical system. So organisms are classified into hierarchy of categories from broad to specific. Broad to specific means plants बहुत बड़ा है. Yeah, if I talk about living beings, so under living beings, plants and animals both will come, right? Plants, animals both will come. Bacteria will come, fungi will come, protozoa will come, right? All of it will come if I talk about living beings. So it is a broader classification, okay? So one of the broadest classification is domain. Domain is a very broader term. Under domain we have kingdom. Under kingdom we have phylum. Under phylum we have class. Under class we have order. Okay, these are keywords, okay, related to taxonomy. Under order we have family. Then under family we have genus, and then we have species, okay. Under family we have genus, species, like that. So broader to specific, okay. So the smallest unit of classification is species, okay, species. So now they are classified based on taxonomic ranks also, okay, taxonomic ranks. Each level in the hierarchy represents a taxonomic rank. For example, the domain is the highest rank encompassing all life forms, domain. If I talk about living beings, it means I am talking about a domain of living beings, okay, it's a very broad term. Then species is the lowest rank representing individual organisms capable of interbreeding, okay, individual organism. So there is species. So a binomial nomenclature has also been proposed. Binomial means only two, right? Binomial nomenclature means how do we write the names of these species? Nomenclature means how do we write the name? Is there any proper way of doing it? So this binomial nomenclature has been given by Linus. He said that genus name and then species name must be used together. Genus name and species. Genus is a broader term as compared to species. So first genus name will come with capitalized first letter, like Canis lupus. I mean, like, you know, Canis lupus, kind of. Agar, for example, dog. Dog is a kind of category. There can be different types of dogs. Jaise, like, can you tell me any name of dog? German Shepherd, aisa. There can be other. Okay. So dog, German Shepherd. Dog, this thing, maybe a different type. Okay. So similarly, so he said, like, although we are talking about the same species here, but he says that genus name must be first, then species name, lowercase, okay, Canis lupus. Canis, then some other name will be there, kind of. Okay, it is binomial nomenclature. How do we write the name of these species, okay? Then if I talk about systems of biological classification, so there are multiple domains. One is 3-domain system. You will be able to understand it clearly, okay, 3-domain system. So it was proposed by Carl Woese. I think his name is Woose. Okay, Carl Woese. Proposed by Carl Woese in late 20th century. This system classifies life into 3 domains based on genetic and biochemical differences. Okay. One is archaea, bacteria, eukarya. Archaea. Archaea means single cell organisms that often thrive in extreme environments. Okay, single cell organisms that thrive in extreme environments. Then bacteria, true bacteria that inhabit diverse environments. Bacteria, I mean, like, we have heard about bacteria many a times. Then eukarya, organism with complex cells containing nuclei, including plants, animals, fungi and protists. I mean, like, eukarya, generally multicellular complex cells. They contain nucleus also. Like, among eukarya, there can be plants, there can be animals, fungi, protists, everything. So it is one of the broader type of 3-domain classification, okay, broader type of 3-domain classification. This classification was proposed by Carl Woese, okay, it was proposed by Carl Woese. After this, okay, after this, we have another system. This is known as 6-domain system, okay, 6-domain system. And how 6-domain system? In the 3-domain system, we had archaea, we have bacteria, and third one was eukarya. Right? So over here, what this person has done, third domain has been broken into 4 subdomains. Third domain has been broken into 4 subdomains. So, like, you know, overall we have 6. So archaea, kingdom of archaea, bacteria, kingdom of bacteria, protista, mostly unicellular eukaryotes, algae and protozoa. Then I mean, like, you know, so the third one, which is third one was eukarya. Okay, third one was divided into 4 subdomains. Okay, 4 subdomains. So, like, one of them is protista. So they are unicellular eukaryotes, algae and protozoa, fungi, multicellular organisms like mushrooms, yeast, etc. Then we have plantae, multicellular plants that perform photosynthesis. Okay, plantae means like plants. Okay, and then animalia, multicellular animals that are heterotrophic. Heterotrophic means they eat the foods that are prepared by someone else. I mean, like, food is being prepared by plants, so they are eating. If they are not herbivorous, so they are eating on other animals. Okay, it is like that. So it is further classification of the first thing, okay, like third eukarya. So here you can see Monera, protist, plants, animals, fungi, etc. So this is, I mean, like multiple models are there. 3-domain model is there, 6-domain model is there, 5-domain model is there. If you open NCERT, 5-domain model is written. Okay, huh? Yeah, yes. So this is the thing, okay. So importance of biological classification. Why biological classification is important? Because it helps us to organize the organisms differently for our knowledge and all. Then it helps us to communicate also. Standardized naming conventions allow scientists worldwide to communicate effectively about a species without ambiguity. And it also helps us to understand the evolutionary relationship. Okay. Then it has practical applications also, such as medicine, identifying pathogens, agriculture, developing resistant crops, conservation, like protecting endangered species and all. I mean, like, it does have a lot of benefit. So this is classification, okay. Challenges in classification: genetic diversity, horizontal gene transfer, evolving taxonomic systems. Taxonomic systems means nowadays we say see DNA sequencing is happening. I mean, like basically a lot of new variations are being created naturally also and otherwise as well. So the thing is, like, this taxonomic classification, this also needs to evolve, okay. It also needs to evolve. So these are some of the challenges that we are seeing. Horizontal gene transfer also happens, like in prokaryotes. Horizontal gene transfer can blur the lines between species and complicate phylogenetic analysis, kind of. I mean, like, these are the challenges associated with it. So is it understood, like, what do we mean by classification system?
Yes.
Classification means what we are doing, all the living beings, we are categorizing them into different types, okay. So we are categorizing. Why we are categorizing? So that, like, we can give them names also, so that we can learn about them, understand about them, so that we can communicate about them to people, and also so that, like, we can take steps to conserve them. So there can be many let's say, like, important things associated with it. Are we understanding? That's the thing. Then microbes in human welfare. So we will discuss about that. So before that, let us do one thing. Like yesterday we have discussed many things, like one particular topic was pending. What? Black holes. So today so much biology, okay. So let us come back to that particular discussion. So based on yesterday's discussion that we had, so can anyone tell me what do we mean by singularity? Quickly, like we are quickly going to revise.
Initial point of black holes part, initial infinite density and infinite temperature.
A point of infinite density and infinite temperature. Okay, singularity. Yes, all the matter has been condensed into a single thing. Like when we discussed about Big Bang theory, so we said that Big Bang has happened from a point of singularity, right? So basically in black holes also, when we talk about black holes, let me quickly draw the structure of a black hole so that, like, you can kind of relate with it. Roughly, okay, I'm just making a rough diagram so that, like, you can have a basic understanding of black hole, okay. I don't know whether these kind of lines are there or not. It is giving it a three-dimensional shape, although kind of, let's consider this thing. So what is black hole? Black hole is basically, like yesterday we discussed about space-time fabric under theories of relativity. The general theory of relativity, when we discussed, so we discussed about space-time fabric. I mean, like, so Albert Einstein said that there is a space-time fabric, and then these heavenly bodies are located there, and because of the presence of heavenly bodies, the space-time fabric sees curvature. Okay, curvature happens in a space-time fabric. Yesterday we discussed about there will be a net. On the net, let's say net is there, fabric is there. If we put a ball of 5 kg weight, 10 kg weight, what is going to happen? That ball will, bring curvature in the space-time fabric. So over here, what is happening? Over here, what is happening? Ki there is a black hole. There is something which has a singularity. Singularity means a point of infinite mass, infinite density, and infinite temperature. So what it has done, it was a space-time fabric. So this space-time fabric has been deformed. Okay, it will come like this. Okay, space-time fabric will be deformed in the sense curvature will be there. So this is curvature. And what is this particular point? This particular point is known as singularity. What it is? Singularity. Okay, singularity. What is this? This particular thing is known as event horizon. Event horizon. What do we mean by event horizon? Basically, this is the area which is under influence of this black hole. This area is under influence of black hole. Means if there are any objects in the universe, that is outside this event horizon, so they can be there outside without any disruption. But if there is anything that is moving, okay, anything that is moving towards it, let's say like anything that comes in contact with the event horizon, so it will be pulled by the black hole. It will be pulled by the black hole. Even if a ray of light is passing near it, if a ray of light is passing, so that will also be attracted towards the kind of black hole. So event horizon is the outer area beyond which things are as it is, but like if anything comes under the influence of event horizon, they will be attracted. Even ray of light will be attracted over here. This is known as black hole. And how black holes are formed? Yesterday we discussed about it a bit.
When their nuclear particle is exhausted, means they stop making nuclear energy.
Means after the energy has been exhausted, right? Because of the nuclear reactions that happen, all those materials are going to collapse. Okay, they are going to collapse. That gas is going to collapse, and then it forms a black hole. Generally, big stars are there. Okay, supernova explosion. Big stars are there. Those stars are thousand times bigger, or maybe million times bigger than the sun that we have in our solar system. So when they run out of energy, so they collapse and they form a black hole. Okay, so black hole is like that. Why we call it black hole? Because the thing is like we are not able to read it. We are not able to understand what is there inside black hole, because even ray of light that goes, it gets captured. Ray of light is also not reflected. So how do we understand the thing? We understand about the thing by hearing about it, or by seeing it, or through various means of inference. How do we see it? Some light will fall onto it, it is going to reflect the light, and then it will be visible to us. So this black hole attracts even the ray of light. Nothing is reflected back from here. So we are not able to read about it. So that's why we have named it as black hole. Long back, people did not know what is there inside a black hole. Did not know anything. But slowly and gradually, we are trying to infer it from indirect evidences. Indirect evidences, yesterday I discussed about gravitational lensing. So what was gravitational lensing? Gravitational lensing means like bending of light happens, right, from the original sources, and then when we receive those light, we feel that this light is coming from a bigger body. Okay, so that body, we are inferring that that body is bigger, but actually that body is not as big. It is because of gravitational lensing phenomena. Gravitational lensing happens when the rays of light bends. When the ray of light bends, so gravitational thing happens. Okay, so if you have the notes of yesterday, note I mean like the kind of printed note that I have given. So in that note, I have included this particular thing also, which is black hole. And then I have included gravitational waves. Yesterday we discussed about gravitational waves, so you just can revise about it. Okay, so black holes are among the most fascinating and enigmatic objects in the universe, characterized by intense gravitational pull. So stellar collapse can happen. Massive stars, when they collapse, they exhaust its nuclear fuel, it collapses under gravity, leading to supernova explosion that ejects outer layer. Okay, core collapse. Also, how the black holes are formed? So stellar collapse can happen, merging of neutron stars can happen, supermassive black holes can also be there. Then types of black holes. There are many types of black holes. Basically, it is dependent on how big the black hole is. Stellar black hole, supermassive black hole, intermediate black hole, primordial black hole. Primordial means like very long back, like historical black hole. Historical means primordial, like kind of black hole. Structure, it has event horizon, it has singularity. Singularity is point of infinite mass and density. Okay. Then photon is like photonosphere, ergosphere, rotating black hole. For rotating black hole, this region lies outside the event horizon where object cannot remain stationary due to frame dragging effect. Okay, like that. Okay, so I have mentioned about some of these things. Okay, so basically, have you heard about that physicist who is, let's say, like we equate him with a kind of Newton? Huh?
What is ergosphere?
Acha, what is ergosphere? She's asking what is ergosphere. Okay. See, now the thing is like some black holes, okay, some black holes might be stationary in the sense that it is here itself, but some black holes are rotating. I mean like outside this event horizon, I mean like some materials are kind of rotating. They are rotating in the sense they are not stationary, they are rotating. Okay, materials are rotating. So any object that comes in contact with the ergosphere, so it is not going to be stationary, so it will be pulled, spiraled into the black hole. So that region is known as ergosphere. Okay, so let's say just a minute. Let's say this particular region, okay, I'm just making it smaller in a sense. Let's consider this particular region is event horizon. Okay, I'm just naming it. This internal region is event horizon. Okay, event horizon means this is the place, event horizon means this is the place where this black hole has highest capacity of attracting the things. Highest capacity means if something comes in contact with event horizon, so they will definitely get pulled. They are going to get pulled by the black hole. Means even if light rays come over here, they are going to get pulled, right? So I mean like this is the place where utmost or extreme attraction is there by the black hole. That's the thing. But outside this black hole, there can be a region where materials are moving, kind of, they are not stationary. This material is not, it is moving. So anything that comes over here, they will also be drifted slowly and gradually. It is going to be drifted, and finally it will come to the event horizon and fall back. So this region where material of the black hole is drifting, it is known as ergosphere. Okay. I mean like event horizon is not the outer boundary of the black hole. Okay, it is not the outer boundary. Slowly and gradually the influence reduces. There will be a place where it has highest influence. Then as we move out, the influence will reduce. Okay, it is like that. Are we understanding this part? So this is known as ergosphere. Ergosphere for rotating or curved black holes. This region lies outside event horizon where objects cannot remain stationary due to frame dragging effects, like that. Okay, so like if we talk about this black hole, I mean like there are many such things, but we just need to remember few important keywords. Okay, what is singularity, what is event horizon, what is ergosphere, and there are many other terms also, accretion disk, okay, gravitational lensing, okay. And so we have discussed about many things, so I just wanted to discuss this thing also, so that like it does not remain undiscussed. Okay, so you just need to read it a bit more. And then gravitational waves is a chapter that we have included here, kind of. You can read that gravitational waves also. We discussed yesterday. So how gravitational waves are generated? When two black holes merge, when there is a kind of a stellar activity of explosion, merging, kind of when black holes merge, they give rise to gravitational waves. And how gravitational waves can be detected? LIGO and Virgo detectors. I mean, like laser interferometer gravitational wave observatory. It's an observatory that detects gravitational waves, right? Virgo is also an observatory that detects gravitational waves. So that way we detect it. Okay. So like when we got to know that gravitational wave exists.
2016 में confirm हुआ और पहले एल्बर्ट आइंस्टाइन ने predict किया था.
हाँ, Albert Einstein had predicted in his general theory of relativity, 1915. Okay. And in 2015-16, it was experimentally identified that gravitational waves exist. And later on, those people have been awarded with Nobel Prize in Physics. Okay. So Nobel Prize, I mean, like, Sir Alfred Nobel. So he was a person, basically, I mean, like, he himself was an inventor. He has invented dynamite. Okay. So that time dynamite was used for constructing roads in kind of hilly areas and all, by how blast and all those things happen. He was one of the greatest inventor of his time, and not only dynamite, he has done so many things. So he considered himself as an inventor. But there was an incident, I mean, like, one of his brother, his name also ended with, started with Alfred, like, Alfred Nobel, Alfred this. So he went to, I think, France, and he passed away in France. He died in France. So next day or later, when he himself visited France, then he saw in a newspaper that, like, so in the newspaper, people have mentioned that the kind of, merchant of the death has passed away. Merchant of death have passed away means मौत के सौदागर की मौत हो गई है. Kind of, someone who deals with like killing of people and all, because dynamite can kill people and all, so it's a blasting thing. But like this has affected him. This has affected Alfred Nobel. He felt that, like, I'm an inventor. People should take my legacy positively as an inventor, not negatively. So like he was one of the wealthiest person of his time. So he said that after my death, right, a foundation should be created where those people who are inventors, those people who are scientists, so they should be awarded, they should be recognized for their contribution. And after he passed away, he has written a will also. Okay, so after he passed away, I mean, like, based on his will, a foundation was created, Nobel Foundation, and yes, in Sweden. So he was a Swedish person, and since then, I mean, like, from 1901 onward, I think, so they started giving Nobel Prize to people. It's been more than 100 years or 125 years. Nobel Prize is being given in multiple fields. Nobel Prize in Physics, Nobel Prize in Chemistry, Nobel Prize in Medicine, Nobel Peace Prize, Nobel Prize in Literature. Then later on they have created another another prize similar to Nobel Prize, but it is being funded by a bank. It is known as Swedish Risk Bank for Economic Prices. Okay, like, so it is equated with Nobel Prize, but like the money does not come from Nobel Foundation. Okay, kind of that thing. So he had huge wealth, and that wealth is like kind of is still there. Kind of it is utilized by the foundation. Kind of investment also happens. So people who receive Nobel Prize, they got kind of, they are recognized also, and then they receive money as well. I think that is kronor. The currency of Sweden is kronor, kind of. And Sweden is a monarchy, kind of, king is there. Okay. So that's the thing. So any question you want to ask? क्यों कल क्या है? किस चीज़ के?
शेड्यूल में तो
नेक्स्ट वीक पे 2-3 क्लास है बस। शेड्यूल में 2 ही क्लास है, बाकी 3 दिन ऑफ।
सिर्फ पॉसिबल है
I don't know, matlab, what I know is that I have to take 5 lectures and I was mentally prepared that I will take classes regularly. हाँ, मतलब I am mentally prepared, but like you told me कि कल क्लास नहीं है.
हम लोगों ने कहा था ये बात।
हाँ?
हमने कहा है कि इसे अगर कर दिया जाए।
हम लोग का इच्छा है कि
5 ही हैं 2 इस हफ्ते में और 3 अगले हफ्ते में, 5 कंटिन्यू कर लीजिए।
अच्छा. Actually, see, like I am not the person who can make decision related to this. Okay, I mean like so director sir is there. Let me check the schedule. Okay, I'll check the schedule, and if there is any change in the schedule, whatever is the case, we will get it notified. Okay, means like I am not the person who takes decision. Are you understanding?
Yes, sir.
So I'll see with them, and if possible, so we will continue the discussion so that within like this week itself the classes are done. Okay, I mean like these classes will help you to understand the thing around science and technology. It's not everything, it's not nothing also, it's like somewhere in the middle, so that like you understand the things, you read from the notes being provided, and then you augment this note, add up value to these notes from the current affair or your own reading. Okay, so that way it will be beneficial. All right, so thank you so much everyone. So Sri Naiki, thank you so much. Yeah.